So I briefly talk about the ocean's role in regulating Earth's climates, but also provide a very important service to human beings, right? So we get a lot of food and energy source, oil, gas, also for recreational activities. We usually go to the beach and however, the ocean is experiencing dramatic changes. Not is largely due to human activities. For example, the ocean is getting a lot of extra heat and it's getting warmer and it's becoming more acidic and losing its oxygen. In the polar areas, ice and glacier are melting, which will affect the sea level and also the ocean circulation, right? So those ocean changes will have substantial impacts on the marine life, biogeochemical cycling, and eventually the ecosystem service for human. So it's really important to evaluate the driver, the mechanism, the rates, and the impact of ocean changes. So my lab is really interested in studying the ocean changes, particularly the biogeochemical cycling, with a focus on nitrogen and carbon cycles. So we want to understand how those biogeochemical cycling are affected by anthropogenic activity, like fossil fuel combustion or fertilizer application, and how those biogeochemical cycling provide feedback with Earth's climates. So see greenhouse gas emission and uptake. Also, internally, the biogeochemical cycling interact with each other. So to study ocean changes, my lab apply a variety of interdisciplinary research tools, such as stable isotope. We use those isotopes to measure how fast those rates, how fast those processes are occurring in the ocean. We're also using molecular tools to identify the microbes responsible for those processes. And we collect most of sample data going to fieldwork and as on research cruises, but we also rely on remote techniques like biogeochemical algal floats and satellite to get a larger spatial and temporal scale of ocean changes. And today I'm going to talk about the interaction between nitrogen and carbon cycle, nitrogen and oxygen cycle. So the oxygen is really fundamental to life, but the oxygen is not uniformly distributed across the global ocean. And as you can see, a lot in the Eastern Tropical Pacific, Atlantic, and Arabian Sea, those regions are shown the reddish color, meaning the oxygen is pretty low. Those regions are called oxygen minimum zones, OMZs. And in addition to those heterogeneous oxygen distribution, ocean is losing its oxygen. And you can see the red color representing in a large portion of the global ocean, there's a decline in oxygen. Over the last 50 years, the ocean has lost around 2% of its ocean content, oxygen content, largely due to warming and changing in stratification, circulation, and other process. But ocean deoxygenation is not limited to open ocean. Coastal water is also experiencing deoxygenation. And so these red dots on this map showing where these hypoxia events have been reported globally. And those hypoxia events meaning the oxygen level is below 2 milligrams per liter, or roughly 60 micromolar. So you can see those hypoxia events have distributed globally, widespread globally, including US, Europe, and East Asia. So one of the main drivers of the hypoxia in coastal waters is excess nutrients, right? So that the excess nutrients, largely due to human activities, will induce eutrophication and drive intensive algal blooms. Then the algal bloom produce a lot of organic matter. When organic matter sink into the bottom, and it will consume oxygen during organic matter remineralization. So when the oxygen consumption cannot be catch up by oxygen supply, and so there will be loss of oxygen, or eventually hypoxia or even aloxia can develop. Right. This is more obviously during the stratified season, in the summer, where the oxygen supply is limited by physical process. So there's actually a very strong linkage between oxygen and nitrogen. If you look at, there's generally a positive relationship between the maximum hypoxia area and the total amount of nitrogen loading into some coastal regions. Those are some of the major coastal hypoxic regions, including Baltic Sea and Northern Gulf of Mexico and Chesapeake Bay. So those hypoxic events will have substantial impact on ecosystem health that can lead to dead fish and bleached corals, right? So we can see the excess nitrogen will induce potentially deoxygenation, but on the other hand, the oxygen will also influence the microbially driven nitrogen cycling process. So there will be a feedback in between. So I'm just briefly showing how the oxygen influence the very simplified nitrogen cycle. So under anaerobic oxy condition, ammonia can be oxidized to nitrite. It's in a process called ammonia oxidation. And meanwhile, nitrous oxide is being produced as a byproduct. So nitrous oxide is a very powerful greenhouse gas. It contributes to around 6% of radioactive forcing. It is the dominant ozone depleting agent in the atmosphere. Nitrite can be oxidized to nitrate. It's called nitrite oxidation. Under anaerobic aloxic conditions, nitrate can be reduced to nitrite, nitrous oxide, N2 in a process called denitrification. It is also an anaerobic organic remineralization process. Lastly, nitrogen fixation can convert N2 gas into ammonia. So previously, there were very distinct separation of anaerobic and aerobic process by oxygen. And so people have investigated oxygen sensitivity of different nitrogen cycling process. For example, the ammonia oxidation on the y-axis show really high affinity of oxygen, meaning it can reach its maximum potential within only a few micromolar. Right, the half saturation constant of ammonia oxidation is around 300 nanomolar. It's very high affinity. And in contrast, for denitrification, it requires lower oxygen. So this figure showing the different steps of denitrification were influenced by oxygen differently. So the first step of reduction to nitrite is less sensitive to oxygen, right? It can occur at high oxygen, but sequentially, last step of denitrification, it can only occur with very low amount of oxygen. So it's a different sensitivity across the denitrification steps. But all of those measurements are coming from open ocean oxygen minimum zone or from a single bacteria culture. So it doesn't really reflect what's happening in the natural coastal environments, right? So the question I want to address is how will marine nitrogen cycle respond to coastal deoxygenation? And will the coastal ocean respond differently to deoxygenation compared to the open ocean? Right. To address those questions, I conducted field work in the Chesapeake Bay, which is the largest estuary in the United States. And it's located east coast and is around DC area and surrounded by Maryland, Virginia. So Chesapeake Bay developed seasonal hypoxia in summer. And this is a model prediction of bottom water oxygen in summer. And you can see in the middle channel, there's dark bluish color represents lower oxygen concentration or hypoxia, even aloxia develops. So in 2022 August, I went there and collected a sample of CB1.5 and CB2 to study the oxygen sensitivity of nitrogen cycling process. And I'm just going to show you the profile of CB1.5 and the result from CB1.5. And the data from CB2 also confirms the results from CB1.5. And so that's a typical profile of estuary regions, warm water in the surface, colder in the bottom water, and fresh on top, and become saltier in the bottom, right? It's very strong stratification, but notice there's not, the water is pretty shallow, it's not 25 meters. If we look at the oxygen profile and nutrient profile on the right, so oxygen shown in the black line, it's rapidly decreasing with depth and become almost aloxic below 15 meters oxygen from 150 micromolar to 0. And ammonia really accumulates in this aloxic bottom water. Nitrite showing in the yellow dots are high in the surface and decrease with depth. And nitrate basically is depleted throughout the water column. So we picked the 3 depths: oxic surface, middle oxic line, and aloxic bottom water to assess the response of nitrogen cycling process to deoxygenation or oxygen concentration changes. The reason we picked those 3 depths is we want to capture different types of microbes that living in those oxygen across oxygen gradients. So the way we use approach we used to study those processes is called N15 tracer incubations. So which my lab really specialized in. So what we did, we're going to focus on 2 processes in ammonia oxidation and denitrification. For ammonia oxidation, we were adding N15 labeled ammonia to the incubation vial and track the production of nitrites and nitrous oxide. Because in the natural environment, most of the nitrogen in the form of N14. When we add in a tracer, it can really enhance our detection sensitivity. For denitrification, we were using N15 labeled nitrates and to track the production of nitrites, nitrous oxide, N2. So this is a photo I was doing the incubation research cruise, and I was doing tracer incubation in those externers, those incubation vials. I was purging, in order to manipulate the oxygen, I was purging those vials with a mixture of helium and oxygen. Right? See, if I want to obtain 0 oxygen, I was only purging with helium. And if I want to slightly increase oxygen, I'm increase the flow rate of oxygen. And I was using the oxygen sensor to monitor oxygen concentration change in real time. So now I'm going to show you all the results. And first, let's look at the ammonia oxidation. Right? So I'm going to show you those plots a lot, so also to talk. Y-axis is the rates, x-axis is oxygen. We have 3 different depths, and the black arrow showing was the ambient oxygen concentration. So if you look at the surface, oxygen surface, it starts from over here, right? That's in situ rates, which are around 700. When I decrease oxygen, it didn't change much. But when I remove oxygen further, it rapidly inhibits. Similarly for the middle oxic line, when I decrease oxygen, it inhibits ammonia oxidation. Interestingly, for the aloxic bottom water, right? In the in situ condition, there's no ammonia oxidation occurring. But when I was adding oxygen to it, it quickly being activated. So ammonia oxidation starts to kick in. This is quite interesting because it looks like those microbes that can conduct ammonia oxidation are living in those apparently aloxic conditions, right? So when you give this oxygen to it, it can quickly respond. Then we fit the response curve using those mechanism type kinetics, and to get this half saturation constant, you can see this key value varies with depth, right? It's much higher in the aloxic water. Then if we look at the production of N2O across different oxygen, you see the aloxic surface. When I remove oxygen, there's rapidly ramp up of N2O production. When I decrease oxygen further, the N2O production decreased. Similarly for middle oxic line and aloxic bottom water, right? So it looks like there's a peak of N2O production across oxygen gradients. This is really being observed in the open ocean. So to better illustrate or to explain this pattern, we define a factor called N2O production yield, meaning how much of the N2O, how much of the ammonia oxidation goes into nitrous oxide rather than nitrites. And when we plot the N2O production yields as a function of oxygen, you can see the black dots really starts to show higher value with amount very low oxygen, right? Low oxygen enhance NTO production yield from ammonia oxidation. Then we can actually fit a function to it to simulate this response. So this is really useful because we can potentially model the production of NTO from ammonia oxidation. So you have already seen this mechanism function. Ammonia oxidation is a function of oxygen, increase with oxygen, and NTO production yields inversely is being enhanced at lower oxygen, right? So if we multiply ammonia oxidation by NTO production yield, so we can get this NTO production rates across oxygen gradient. So that's the NTO production rates across oxygen, right? You can really see this peak at certain oxygen levels. And so this is really useful because we can incorporate into biological model to estimate NTO production in the coastal waters. And actually that's what my lab is doing with colleagues from WIMS and GFDL to implement those parameterizations. So now we have talked about Chesapeake Bay or estuaries, but how does it compare to the open ocean observation? So here I'm showing the ammonia oxidation as a function of oxygen, but I'm slightly changing the y-axis. I'm showing the relative to maximum. Because the rate vary across different depths, I want to show the rates on the same scale. I've just normalized the rate by the maximum rate that I observe at each depth, right? So you do see it's increasing with oxygen. And similarly, we fit this response curve. But let's look at data from open ocean of oxygen minimum zone, right? Eastern tropic Pacific or South. The red dot looks like reached its maximum at slightly low oxygen, right? Meaning it's more sensitive to, it can grab those oxygen and do ammonia oxidation on low oxygen. If we fit a response curve to it, right, that's how it looks like. See, so this is quite interesting because see if the oxygen is losing in this water, see if oxygen is at 10 micromolar, and ammonia oxidation in the Chesapeake Bay or in estuary is only 60% of its maximum. But in the open ocean oxygen minimum zone, it can still above 90%, right? That shows the difference. And we were hypothesizing this might be due to the microbial community inhibit in the oxygen minimum zone versus in the Chesapeake Bay. I'm not showing the data, but we are finding in the Chesapeake Bay is dominated by ammonia oxidizing bacteria, but in the open ocean, it's mostly ammonia oxidizing archaea, right? So it's some work we're still working on. So we have talked about ammonia oxidation, and let's jump to the nitrogen removal process, the denitrification. Right, so denitrification is unique as 3 different steps: nitrate reduction to nitrite, nitrite to N2O, and lastly is N2 production. And if you look at those response curve, the first step of denitrification for the oxygen water, when I remove oxygen, the process increases rapidly. Similarly for middle depths and for the aloxic bottom, when I add oxygen to it, it inhibits denitrification. Right? But also very interesting, meaning if you look at the surface oxygenated water, the denitrifier are present. Right? When you remove oxygen, they start to reduce nitrates. Right? And then we fit those response curves showing the dashed line, use this exponential decatur. So we can get this key value, meaning the rates reach around 37%, basically the e-folding of these maximal rates. If you look at the 2 other denitrification steps, they're quite similar. The denitrification was really enhanced at lower oxygen. But one thing to notice is the rates is quite different, right? In the surface, the rate is much lower, and aloxic bottom water is much higher rates. So now I'm going to just combine all of those data and showing those normalized rates. So that's the normalized rate for nitrate reduction to nitrites, reduction to N2O, and reduction to N2. Right? So if you look at those response curve, they become basically not present when oxygen is above 10 micromolar across all different steps. When oxygen is below 5 micromolar, this starts to kick in, right? Higher rates on low oxygen. And if we fit this curve to it, and this K value is about 1 micromolar, it's very consistent. But if I introduced this at the very beginning, if you look at those culture experiments, right, their response curve. And so the first step of denitrification can occur at much higher oxygen. And so their oxygen sensitivity vary across different depths, across different steps, right? Unlike in the Chesapeake Bay, they're just very the same. And this is also people observing in the oxygen minimum zone. So the data from oxygen minimum zone are shown in the reds, right? The first step is not that sensitive to oxygen. It can occur at high oxygen. And then it's more being constrained to low oxygen for the second step, and even more so for the last step. So we're thinking that might be due to the presence of diverse denitrifiers, and also the amount of organic matter and nitrates or nitrite present in this water. In the oxygen minimum zone, there are relatively more nitrates, less organic matter, but in the coastal region, you got relatively more organic matter. So we are actually still working on that to characterize the microbe community in those environments and to develop ecological system model to quantify that response. But if we zoom in further, and so most of the biological model only incorporates this last step, the N2 production, right? The denitrification all the way to N2. And we fitted this curve. If you look at Chesapeake Bay, showing blue line, oxygen minimum zone showing in the red, right? You can see the distinction. The Chesapeake Bay denitrification can occur at slightly higher oxygen, but not in the oxygen minimum zone. So just to put all those things together, which I show you the first is the oxygen sensitivity of ammonia station between Chesapeake Bay and oxygen minimum zone. Chesapeake Bay shown in the blue, oxygen minimum zone showing reds. Now let's add denitrification into it. Right. Again, denitrification showing dashed line is enhanced with lower oxygen. Right. And so, but one interesting thing is there's actually an overlap between ammonia oxidation and denitrification. Right. There's no direct distinction or oxygen threshold for where ammonia oxidation can occur or denitrification can occur. And more interestingly is those overlap for the coastal waters at higher oxygen and a wider range, but is really constrained to low oxygen in the oxygen minimum zone. But what does it really mean, right? What's the implication of that? So if we go back to this cycling process, ammonia oxidation converts ammonia to nitrite, and nitrite actually is a substrate for denitrification, right? So if those two processes are overlapping or coexisting, they actually really enhance those cycling process, which really trigger N2 production and N2 production, right? You will enhance the nitrogen removal. So let's just take N2 as an example, see how that will affect N2O production of cycling. So I'm showing the N2 production of cycling rates on y-axis. Positive means net N2 production. Negative means net consumption by denitrification across oxygen gradients. I already showed you that N2 production of ammonia oxidation, there is a peak, right, at certain oxygen level. And denitrification N2 production is really enhanced when oxygen is very low. For the last step of denitrification, N2 reduction of N2, it also becomes dominant at very small amount of oxygen. So that's all the cycling process. If we combine all those three processes together, that's we get those purple line over here, right? And so in the oxygen water, most of N2O is coming from ammonia oxidation. When deoxygenation occurs, denitrification starts to kick in, increase N2O production. But when the oxygen drops to zero, N2O is being consumed. There will be net consumption of N2O. So eventually, there is a certain oxygen sweet spots or hotspot for NTO production. And this really helps to explain why there's variation of NTO across oxygen gradient in the natural environment. And this is also very useful for modeling studies. And actually, we are incorporating those parameterization in the climate model, in which most of the Earth's model, climate model, does not incorporate NTO. And now we just go back to the question I raised at the beginning. Yes, so the deacidification will inhibit ammonium oxidation while enhancing deacidification and NTO production. There is very clear distinction between open ocean and coastal water in response to deoxygenation. The coastal water nitrogen cycling is likely more sensitive to deoxygenation than the open ocean. In that sense, because ammonium oxidation inhibits at a higher oxygen concentration, while deacidification is actually occurring at slightly high oxygen in the coastal water than in the oxygen minimum zones, right? So looking into the future, the oxygen will continue to decline and in the open ocean and also in coastal water due to temperature change, circulation change, and project increase in nitrogen loading into coastal environments. So it's our observed relationship between nitrogen and oxygen cycle can really help to evaluate the future changes of oxygen and nitrogen availability. But that story doesn't end here, because I mentioned the ocean are experiencing many other changes, right? Like the temperature, pH, and glacial melt, etc. Right? And so my lab is continue working on those pressures and trying to better evaluate the compact effects on biochemical cycling process, not only from single effects. But hopefully I can give you updates in future faculty seminars. I will stop here and happy to ask questions. Thank you.
All right, hi everybody. So today, I guess I'll say thank you for considering me still new. I guess I would say I'm new-ish. So I'm starting my third year. And so today I'm going to be presenting data that has been mostly collected by my first master's student and the REU students that we had over the summer. Maybe. All right, we'll do that. So I'm going to be talking about two stories. The first story is my master's thesis, or my master's student, Lydia Rugel's thesis project. So she's been looking at identifying B vitamin requirements and phycosphere interactions for Pyrodinium bahemense. And the second project that I'll be talking about is untangling impacts of bacterial interactions and changing environmental conditions on this same harmful algal bloom dinoflagellate. And this is the project that the REUs did over the summer. You're doing great. Okay. The left mouse button, I'll try. So in general, my lab is interested in how phytoplankton interact with all other microbes. So throughout my PhD and my postdoc, I have mostly been thinking about how phytoplankton have interacted with other eukaryotes and bacteria, but I am open to all interactions, and I think aspirationally, I've put viruses and fungi on this plot as well, because these are things that I hope to think about in the future. Left button. But for today, we're going to be really thinking about how phytoplankton interact with bacteria. And some of these bacteria can be friends, symbiotic bacteria, and some of them can be a little bit more opportunistic, where they might be taking advantage of the phytoplankton in some situations. And as I said, we're going to be talking about Pyrodinium, which is a harmful algal bloom dinoflagellate, and this is Lydia's work that we're starting out with. So a little bit more about who Pyrodinium is. Pyrodinium is a dinoflagellate that is able to produce saxitoxins. Saxitoxins are extremely potent neurotoxins. So if these are bioaccumulated in shellfish and then consumed by humans, they cause respiratory failure and death. So in the Florida region, these have been accumulating in scallops. This year, the scallop season was terminating terminated early because there were saxitoxins detected off Pasco County. This also happened last year. In the Tampa Bay area, we're not really eating from Tampa Bay very much, so we're more concerned about how the blooms are influencing overall water quality, and there's some connection between these blooms and the degradation of seagrass beds in Old Tampa Bay. So there's a lot of interest in thinking about what is causing these blooms and which factors can be used as predictors. So over time, there have been some correlations between things that are common in bloom-prone areas. And some of these include really shallow estuaries that have high residence time. So if you think about Old Tampa Bay, there's a really high residence time there because of all the bridges. Generally, we have blooms occurring in eutrophic estuaries that in Florida have low and variable salinities, but in the Caribbean, where the Pyrodinium also blooms, they like really high salinity. So 40 PSU, so there's a little bit of inconsistency there. They generally like it warm and they generally like it rainy. So in Florida, this has historically been these blooms have been strongest in these three regions, but this is historical, and the last couple years have been anomalous. So the last 2 years have had no blooms in Old Tampa Bay, and a lot of additional blooms along the Gulf Coast in other estuaries that haven't previously seen blooms. So typical bloom drivers are not consistently correlated with cell abundance, and as a result, a predictive framework for these blooms have so far remained elusive. And this led us to think about some other factors that might be contributing to bloom formation. So when your normal players like nitrogen, phosphorus, temperature are not really good predictors, sometimes there are some kind of more enigmatic factors that are contributing to bloom formation. So we started thinking about micronutrients, which can be such as vitamins or trace metals, can be really critical growth regulators. And we know that a lot of harmful algal bloom dinoflagellates do require B vitamins for growth. And we also know that there have been some instances where the bacteria that live on or near phytoplankton have been shown to specifically provide B vitamins that allow those phytoplankton to grow. So with these things in mind, we hypothesized that vitamin B12 availability and bacterial interactions are influencing Pyrodinium bloom formation in the Florida estuaries. And we set out to characterize first the bacteria that live with Pyrodinium, and then also how much and which B vitamins Pyrodinium needs in order to grow. And then finally, we wanted to see if we could show that bacteria actually provide these vitamins to the phytoplankton. So we had 2 strains of Pyrodinium that were provided to us from FWC. One was from the other coast, from Indian River Lagoon, one was from Old Tampa Bay. And these were both originally isolated from the cyst form. So when they are upset for any various reasons, they might form these resting cysts that can hang around in the sediments for quite a while. So these were isolated originally as cysts, and then they were grown up into the vegetative state to produce these nice, clean cultures that we were working with. So to first think about what we're starting with, we looked at the microbial composition of the communities in these cultures. So here is the microbial community composition in the Indian River Lagoon, the IRL culture. And we can see that there's some variability here. Overall, there are about 20 ASVs, so these are like strains-ish of bacteria. And that is very different than what we saw in the Old Tampa Bay cultures, where we only have these 3 color blocks, and that was a total of 6 strains. So this 20 is much more typical for phytoplankton, and the groups that we see in this Indian River Lagoon culture are pretty normal bacteria that are found over and over again in phytoplankton cultures and also in surface seawater. When we looked at these bacteria a little bit more closely, we found that these were actually bacteria that are typically associated with sediments, and and even more specifically, pretty polluted sediments. So we have different starting materials that we're working with, and we thought that this old Tampa Bay culture could be a really nice opportunity to do some experiments because it doesn't have that typical phycosphere phytoplankton bacteria interaction association already established. So going forward, we basically worked with this old Tampa Bay strain to do our experiments. So we grew up the old Tampa Bay strain with that low diversity microbiome in media that was missing one of the 3 B vitamins that are most commonly given to phytoplankton to help them grow. So that is B12 or cobalamin, B7 or biotin, and B1 or thiamine. And then our positive control is the replete with all 3 of those vitamins available to the phytoplankton. So we have 4 serial transfers, sort of. So for the minus biotin, the minus thiamine, and the replete conditions, these are serial transfers. And in each case, the growth curves are pretty robust, and that gives us evidence that the phytoplankton are able to grow without biotin and without thiamine, even after those 4 serial transfers, so that eliminates the opportunity that there is leftover luxury uptake. So this is really good evidence that they don't need those vitamins in order to grow. The exception there is the cobalamin. So as soon as we took away the cobalamin, they immediately stopped growing. And this is a little bit unusual for phytoplankton. Usually it takes at least 1 transfer for them to use up what they have left over, stored in the cells, or like leftover vitamins that are in the transfer media when you create that new culture. So this gives us the indication that they probably need a lot of B12 in order to keep growing. So the next thing that Lydia did was to do some serial dilutions of B12 and grow up the pyrodinium in these different concentrations of B12. And so you can see there's kind of like a dose response curve here where the amount of total growth increases with the amount of B12 available to the pyrodinium. And if we can compare these values to, oh, sorry, I got ahead of myself. So this is a correlation between our chlorophyll relative fluorescent units and actual cell counts. And so if we use this line to connect these 2 values, we can say that this 2000 RFUs is approximately bloom level in Old Tampa Bay. And so in order to get above bloom level, we need more than 3.69 picomolars of B12. And we can then compare that to other phytoplankton species that are also saxitoxin producers. So we have Gymnodinium, Alexandrium, and another species of Alexandrium, and they also have pretty high B12 requirements. So we have 1.52 picomolar and 0.34 picomolar. And then finally, we also compared this to Karenia brevis, which only needs 0.01 picomolar of B12. So in general, we're seeing a increased need for B12 among saxitoxin producing dinoflagellates. So the next thing that we tried is, so we know that our OTB, our Old Tampa Bay isolate, has really low diversity microbiome, probably not a typical phytoplankton vegetative cell microbiome. Can we add bacteria back into that culture and then rescue its ability to grow without B12? So we had 3 sources of bacteria. The first was from the Indian River Lagoon culture, that one that I showed you at the beginning that had the more typical culture microbiome. And then we also took bacteria from the Old Tampa Bay sea surface and Indian River Lagoon sea surface. And one thing that I want to point out here is that at the Indian River Lagoon, when we collected this sea surface water, there was a pyrodinium bloom occurring at that time. Whereas in the Old Tampa Bay sea surface water that we collected, there was no pyrodinium growing at that time. And then we did some pretty aggressive filtering of these different sources to try to remove all of the eukaryotes. So for the cultures, we have the culture source for the bacteria is going through the 10 micrometer pore size and the 0.8 pore size filters. For the seawater samples, we added another filtering step because we were having some very pesky, very tiny diatoms sneak through our filters and ruin our experiments. So I think it's important to kind of be transparent about the fact that we're removing a lot of the bacteria in these cultures or in these sources. So this is not giving them the opportunity to recruit anything that's out there. We are really diminishing their recruitment pool by filtering this way. And we can look at this too through flow cytometry. So this is the raw Old Tampa Bay seawater. And then this is what it looked like after we filtered it. So we're removing a large proportion of the cells that are in the seawater before we're giving it to the pyro to have that opportunity to recruit new microbiomes. So these are the pyro growing in pink is replete, and then green is minus B12. On the left is the supplemented with the bacteria from the Indian River Lagoon culture. In the middle is supplemented with the bacteria from the Indian River Lagoon seawater during that Pyrodinium bloom. And we can see that this is kind of the only case where the Pyrodinium does have a little bit of growth when there's no B12 present. Not a lot, right? It's not reaching all the way to the same levels as when we give it a whole bunch of B12 in the media, but we are rescuing a small amount of growth here, and this was sustained through a serial transfer. So there's something in this bacterial community that we gave it that is able to provide at least a small amount of B12. So we at this point, we filtered all of these cultures down to look at what bacteria we actually ended up transferring there and to see if any of the bacteria in particularly this one are B12 producers that might be symbiotic to the Pyrodinium. And as a reminder, that 2000 line is the bloom level. So the bacteria and the growth that we're seeing in the no B12 conditions is still not reaching anywhere near the bloom condition. We're just seeing a little bump in growth that is interesting and exciting for us. Okay, lots of colors. So the main thing that I want you to see in this plot is that this is recruited. So these are all of the cultures that were given the opportunity to recruit bacteria from the Indian River Lagoon seawater. These are the communities in the cultures that were recruited from Old Tampa Bay seawater, and these are the communities that were recruited from Indian River Lagoon culture. And the main point here is that there's a lot of diversity. Like, we did it. They recruited bacteria from the sources that we gave them. They no longer have these very minimal, tiny, low diversity communities. So now that we know that we were able to transfer bacteria, we want to start looking at particularly what's special about this one. What did they recruit here that is different than all the other ones that might be providing the B12 for them? This is actually data from another experiment. This is from the summer with the REUs. And this is just to show you that it's not just the diversity that's changing, it's the overall biomass of the bacteria in the cultures. So this is experiment day on the bottom, and then this is bacterial cell concentration. And the low diversity microbiome, the one that we started with, it's not only low diversity, there's just not that much growing. But when we add the Old Tampa Bay seawater bacterial community, we're increasing the diversity, and there's a lot of bacteria that are actually growing in the background. Okay, so what's different? Some of the things that I picked out from looking at these plots are the Enterobacterialis and the Chitinophagialis as being things that are kind of only present in the Indian River Lagoon seawater and not in the other groups. So we can dig into that a little bit deeper. And these are 3 genera of Enterobacterialis that are known B12 producers, and they are enriched in the Indian River Lagoon transferred microbiome. These are also bacteria that probably came from a human source. So these are things that will come from wastewater treatment plants. These are things you don't really want to eat or drink or have in your environment, but they seem to potentially be doing something positive for the phytoplankton. The Chitinophagialis were a little bit more clear in their pattern, where they were only in these, where the source was coming from Indian River Lagoon and there was growth without B12. But this genus, Luinella, and then obviously this is just an uncultured group. We don't know anything really about whether these are able to produce vitamins, unless someone else knows. I don't. And the literature wasn't able to tell me anything on that, but they are groups that are commonly associated with phytoplankton. So this isn't the first time that they were found to be associated with a phytoplankton or macroalgae. And then Alteramonas, I think, is also an interesting one to think about, but in the opposite side of things. So Alteramonas cannot produce B12, cannot, and also generally needs it in order to grow, or needs it to grow well. So it can sometimes grow without B12, but in order to grow really quickly, it needs to have B12 in the media. And it's only really growing in the Indian River Lagoon supplemented community. So this is another indication that there is some B12 being provided here from another bacteria. So we're probably having these multipartite interactions where the Alteramonas is also uptaking some B12 that's being produced by another organism. So from this first part, some conclusions that we can come away with is that Pyrodinium is very clearly oxytrophic for B12, but not for the other B vitamins. It seems to require more B12 than other Hab dinoflagellates, like our kind of most infamous Hab group in Florida. It needs a lot more than Karenia brevis does. And the bacteria that is recruited from the Indian River Lagoon seawater that was collected during a Pyrodinium bloom seems to be able to support some growth, but it may not be able to produce full growth, potentially because we've filtered out so much of that bacteria in the starting community that we're losing some of the more important groups. And we're also thinking about whether or not there might be some additional sources, such as from just directly from wastewater, not from bacteria from wastewater, but from human excretion. I don't know if you've ever looked at the ingredients of one of your Celsius cans, but it's something like 10,000 times daily required needs for vitamin B12. And vitamin B12 is a water-soluble vitamin. So there is a strong chance that there is an increased B12 loading from human sources with current trends in nutrition. So some of our kind of future and current directions that we're working on is to, we're partnering with Old Tampa Bay, and our new graduate student Liliana is taking on this project. We are collecting seawater from throughout Old Tampa Bay, and we are doing organic phase extractions and using high resolution mass spectrometry to look at the different types of B12 in all of these samples to try to identify B12 sources. And then at the same time, we're looking at microbial communities here and potential microbial B12 producers in different regions of the bay. And I have a whole other project that I wanted to tell you guys about that I'm just going to introduce because I talked about the first project for too long. So the other project that we've been working on in the lab, this is still pretty preliminary, but the RUs and the rest of the grad students in the lab have been working really hard on this project. It was very labor intensive, so I need to at least mention it. So we've been thinking about untangling bacterial interactions and changing environmental conditions on the growth and toxin production of Pyrodinium. And so instead of just thinking about bacteria, we're thinking about how bacteria and environmental conditions interact and then feed back on Pyrodinium growth. So we know that there are changing climate and weather patterns, right? I hope we're all in agreement about that. So we have increasing temperature and changing precipitation patterns, where if we have a really hot summer with high drought conditions, we're going to have higher surface water temperatures and higher salinity due to evaporation. Alternatively, we can have higher temperature conditions with more acute rain systems where we have reduced salinity, at least locally or for short amounts of time. And kind of tangled up in this is that when we have increased rain, especially when we have those really high volume rain events, we can have increased nutrient loading. But all of these things are happening at the same time. So it's really hard to think about which thing is most important and which part of this system is having the most control over the growth of these different organisms in the bay. So we tried to separate it all out through a multifactorial experiment where we have our two different microbiomes in our cultures. We have different salinity conditions. We have different nitrogen or nutrient conditions, and then we have a challenge temperature, a hot temperature, 32 degrees Celsius, and 24 degrees Celsius is on the lower end of temperatures that you would see in the summer. Maybe you wouldn't see those summer temperatures in the summer, but this is the kind of like the base culture conditions that we're working with when we're just maintaining these cultures in the lab. And so if we have all of these things, all these different conditions, and we have it all in triplicate. We have 72 culture bios that the REUs, with help of Lydia and other people in the lab, were measuring every other day. And when they were measuring them, they were measuring not just the chlorophyll this time, but we were also doing high imaging flow cytometry so that we were looking at the Pyrodinium cell morphology and also the bacterial cell counts throughout this entire experiment. So something that came out of this that was pretty interesting, this was our first trial, and this trial is short. It's only 12 days because this was from last summer, and there was an incident that happened that caused electricity to stop being supplied to our lab for a long time. But we saw something exciting where our low diversity cultures were growing a lot better than the high diversity cultures at high temperature. So this was really intriguing for us, and we wanted to follow that up. And so we redid everything this summer with a lot more time. So we did 2 more trials, and then this was the serial transfer to trial 3. And we did see the same thing where we had our low diversity in the lighter colors. Those cultures did better when it was hotter and higher salinity, and when we had more nutrient availability, compared to the high diversity cultures seemed to do better when it was a little bit colder, and when there was less nutrient availability. So we've done a lot more with this, thinking about the bacterial growth and which bacteria are actually there. There's not quite enough time for me to show you all the results from this project, but I do want to show you some of the images from the flow cytometer. So these are some of the images that we're taking throughout the course of the experiment, and we saw some very interesting morphologies, particularly these two at the end, where we started seeing a lot of cells with these vesicles attached to them. And we started having a lot of cells that were in this sort of apoptotic phase that were, honestly, we don't know. These are either dying or they're undergoing sexual reproduction. And it's hard to tell. It's either good or bad. But yeah, so a really cool tool to be able to think a little bit more deeply about what's happening under these different conditions. And so this is a stay tuned type situation. We are working up this data currently, and I probably will be able to give you a lot more conclusions about all of these different interactions next year at faculty seminar. The take homes from that is that so far we're seeing that the Pyrodinium is doing a lot better at low temperatures with their high diversity microbiome and at high temperatures with the low diversity microbiome. And the impacts of the microbiome on toxin production are also to be determined. So, yep, I'd just like to say thank you to all the lab members that have been working on this. And these are some of our funding sources for these works. And yeah, if you have any questions, I think I have time for like a question or two.
You know, it's pretty well established now that iron is really important in the ocean. It's a micronutrient that limits phytoplankton and microbial growth over a lot of the oceans. This is just the model output showing the different limiting nutrients. The light blue is iron, so it's the limiting nutrient over large areas of the surface ocean. So we really do care about how it gets into the ocean. Iron is pretty low in the ocean. It's pretty hard to measure. We didn't really have good data to the 80s and 90s. And really by the sort of 1990s, the prevailing view was that if you have dust deposition to the ocean, you have iron, and that's where you have no iron limitation. So I'm maybe going to call that the Gene Shin view of the world. And this is how we thought of the iron cycle up until about the year 2000, I would say. That said, this is a figure showing all of the dissolved iron water column profiles in 2004. We only had something like 25 depth profiles for the whole ocean. And if you think about some of the EWOS sections and what we have for nitrate and phosphate, this is really low. And this really motivated the Geotraces program to try and increase coverage across the oceans, which started in sort of 2004. I was just involved last year in leading a special issue of Oceanography that's, if you're interested in all kinds of trace elements, this is great to check out, which looked at the sort of findings we've had from the last sort of 20 years. One of the sort of products of this is that we have publicly available products, so you can go use all this data for free. And we now have something like 16,000 measurements of dissolved iron in the ocean that we trust in the latest data product. And this figure shows all the cruises where we have dissolved iron sections. So we're in this nice position where many investigators have measured these sections, and we have a lot of information to try and understand the cycle. I should say that we're going to have a new release at the end of this year, which will almost double the amount of dissolved iron. A lot of people have spent a lot of time putting this together. So if you're interested in any trace elements, this will be coming at the end of the year. So as part of this, myself and a couple of co-authors produced a review of where we are with thinking of dissolved iron in the ocean. This is the sort of accumulation of 20 years of work. And we put this figure in, which is all the Geotraces data for the different oceans, with these light blue colors showing very low dissolved iron, especially in the surface. And then these hot colors showing hotspots of high dissolved iron, especially at depth. We see them sort of close to the margin where there are sedimentary sources, and we see them nicely around hydrothermal venting on the Mid-Atlantic Ridge, for example. So we now have these huge data sets. We're definitely in this position where we can say there are these multiple iron sources. They're really important. It's not just about dust to the surface ocean. That's still important, but we have to understand these other sources. And this is just a very sort of simple conceptual cartoon that makes that point, that these are all the different iron sources we have to think about, not just the atmosphere, but these hydrothermal vents and margin sediments. And these were perhaps hinted about before, but we thought that most of the iron would sort of drop out of solution pretty close to the source. Actually, what we're seeing from Geotraces is very long distance transport of dissolved iron. And coincidentally, I thought if you're not that familiar with hydrothermal vents, our own Jackie Dixon is actually out. I think she came back yesterday at the Endeavour Seamount, driving ROVs and looking at hydrothermal venting. So I stole these pictures from her Facebook. It's pretty cool. Something with iron you can actually see in the ocean. And we can just zoom in on two of those sections to really make that point about transport of iron. That sort of light blue is the background. And if you look at these zonal sections of the South Pacific and the North Pacific, you can see these sort of hotspots that are transported across the ocean. So there is hydrothermal iron traveling thousands of kilometers through the deep Pacific, and then iron from sediments traveling thousands of kilometers through the deep Pacific. So we really want to understand how far these sources travel, how important they are in different places, and ultimately do they get to the surface. Ooh. That's So I'm going to talk about iron isotopes, which is what we do in my lab. Don't know how this has got so mangled, but basically we're going to be looking in Delta 56 iron, which is basically just the difference between a sample and a standard. And there are changes in that number from various geochemical processes. So the hope is that this number will allow us to fingerprint the different sources which have different histories. Now, I put this figure in because this is all of what was known about iron isotopes in the ocean before we did any seawater work back in 2003. There's lots of different things here, but the thing to notice is the two sources to the ocean that they considered at that time was atmospheric dust and hydrothermal venting. So that was plus 0.1, which is basically the crust, and hydrothermal venting had this slightly negative signature. And the reason I like showing this is because they then took this and they modeled the whole oceans from this. It's almost completely wrong because there's so much variability, but I like the fact they tried to do that. You know, the North Pacific all being dust and something over here in the South Atlantic all being hydrothermal. But if you remember what we knew about iron in the ocean in 2000, it was pretty hard to do this. And the other takeaway point is that they don't even include sediments here. So that's what I'm going to talk about. Okay, so we come back to our horribly mangled slide. I want to show you, this is the dissolved iron in a sediment pore water profile. So the water in between the grains of the sediment. And we can see there's a lot of variability in iron isotope signature through just 30 centimeters the sediment or so. That's really good if we want to come up with a sort of nice tracer of sedimentary iron. The other thing to point out is in this blue region, we have these very negative values of -3 to -1. This is in the ferruginous region of the sediments. It's where there's low oxygen and iron is being used for respiration. So iron 2 is being produced and it has this very diagnostic signature. That's really good for us if we want to use it as a tracer. So I'm going to ask a couple of questions that will help us sort of put together the pieces for using iron isotopes. The first of all is, does dissolution of marine sediment impart distinctive signatures? And I've kind of given away the answer to that in the last slide, but we can look at it in a bit more detail. So this is another pore water profile. This is from Eel River, which I always forget if it's in California or Oregon, but on the West Coast. Again, about 20 centimeters, we can see this very high iron concentrations. And this, again, very consistently light or negative iron isotope signature. So that's showing the same thing. In these anoxic pore waters, you've got iron being produced with a very light signature compared to the crust. I'll put that on one side. So we know some things about this mechanism of dissolution. This is a mechanism we've known about since 50 or 60 years or more. It's a very classic way of getting iron production in sediments. We know a few things about it. It's high in iron. Pore waters are anoxic, and it has this very light signature. So we call this reductive dissolution in the sort of iron isotope world. And we also know that really the only way of getting this into bottom order is going to be diffusion of that dissolved iron too. So that's our nice example. We also have a second example that's come about because of iron isotopes. So this is some pore waters from the Southern Ocean. You can see they look quite different. The iron is concentrations are very low, and the iron isotopes are very unfractionated. And so what we think here is a second method of production of iron in sediments. It happens under oxic conditions, low iron. It has this crustal signature of 0.1 per mil. This is something we've been working on. And we call this non-reductive dissolution or lithogenic production. And really what we think this is the result of kind of weathering, producing sort of small particles of crust, which are small enough for us to see as dissolved. But we have these nice two very distinct mechanisms in terms of isotopes. So this is one nice thing that's come about from measuring iron isotopes is we have what we think is a second method of producing what I'm calling dissolved iron in pore waters. And since these are colloidal size, so they're a little bit bigger, they're probably basically small particles, we're probably thinking about resuspension or sediment disturbance in getting them into the water column in, say, nepheloid layers or somewhere. So you might expect them to behave quite differently. Now, the other thing we've been thinking about, and this was led by my collaborator Will Homeke in 2021, is thinking about where these might occur in the ocean. And so this divides into sort of reductive dissolution up here and non-reductive dissolution. What we can see is that we would expect this reductive dissolution, which is oxygen sensitive, to happen where there is high TOC flux resulting in low oxygen. That's probably going to happen on sort of shallow parts of the ocean. It produces soluble iron. And then this sort of non-reductive process may dominate in the deep ocean where there isn't a lot of low oxygen conditions. And we're thinking more about sort of disturbance of the sediments. So we think these mechanisms might dominate in different places. And obviously there also might be places where they both occur. So I'm sort of going to leave non-reductive dissolution aside and talk about reductive dissolution for the rest of this talk. But it's quite exciting we have these sort of ideas. So if we want to use isotopes as a tracer of sediment input in bottom waters, we need to know, does that diagnostic signature actually make it into bottom waters? And does that happen without fractionation? You know, a lot of that iron that diffuses out is going to precipitate, and that could affect our isotope ratios. And here there's actually some really nice previous work done off the Californian coast in these sort of low oxygen anoxic basins, San Pedro and Santa Barbara near LA. Here's a really nice pore water profile again from Silka Sevelman. In these anoxic pore waters, again, you're seeing this -2, -3 signature. What was nice about this location is they also did benthic chamber incubations where you put a chamber down on the sediment and you see what comes out over 40 hours. These don't happen very often because they're hard to do, but they saw that this light signature was persisted all the way through this. So this is showing is it's coming out of the sediments and it's persisting in the bottom waters. Seth John then in one of the first seawater isotope profiles actually saw basically the same signature in bottom waters. So this was pretty good evidence that to some extent this diagnostic signature of the iron 2 is crossing the sediment water interface, at least in these anoxic waters. We've also been doing some work on this, and Hannah Hunt, who recently graduated from my group, has been looking at this in the South Atlantic. This is the same pore water profile I showed you, which is from the Cape margin. And then she has some data from the Benguela upwelling. We can just look really at this one, which is the iron isotopes in iron coming out of sediments. And we see it's about -3, which when we look closely is very similar to what we see in the pore waters. So again, we're seeing this signature across the interface. Here it seems to be happening in more oxic conditions because it doesn't get as low oxygen where this was measured. So this does suggest to some extent that the signature can make it into bottom waters. Okay, so there's still a lot of nuance there and a lot of things we're working on to figure out exactly whether it fractionates to how much, but it seems like to some extent it's faithfully transported across. So the next question is, you know, do they really get transported? Does that sort of -2 per mil or -1 per mil actually get transported through the ocean? Again, without fractionation. So this is what my lab has been doing for the last 5 years. It's been trying to increase iron isotope coverage of the ocean, I guess. These are locations where we had iron isotope profiles in 2021. As we move forward to 2025, we have a lot more iron isotope profiles. We've really been trying to examine some of the Pacific data. So we've measured a lot of samples. And I should say that this is all the work of Matthias Sieber, postdoc in my lab, and Hannah, who actually graduated a week or 2 ago. So I'm going to show some data from this section across here, and then some of this US section here. So this is a figure from Nishioka et al. So it's kind of flipped. So you've got Japan over here and the US over here. What you can see in these dissolved iron sections, just focus on the red being the high iron and the sort of blue being low iron, is that there's a lot of iron coming out of these marginal seas close to Japan, and it's being transported throughout the North Pacific in the North Pacific intermediate water, which is the same section I showed you earlier. So this is a nice place for us to go and look at the iron isotopes and figure out what they're doing in the same place. So this is Hannah's iron concentration section for the same cruise. You can see the same thing, which is good, that you have this sort of hotspot of iron coming off sediments and being transported eastward. And from this cruise, we can estimate that that signature is traveling at least 3,000 kilometers mostly through lower North Pacific intermediate water. Now, if we put the iron isotopes section on for the same cruise, first of all, we can see a lot of blue. That means it's all very sort of negative. The other thing we can see is when we look closely at that plume, it's all very close to -1 per mil. It's very conserved through the plume. So that was a nice finding. It looks like whatever signature is being imparted is being transported all the way through the North Pacific. That's really helpful if we want to trace how far it goes. And we also have some sort of near source sediment information for what's coming out, and that's about minus 1.2. So it seems like what we're expected to come out of the sediments is making it through the ocean. Now we can look a little bit closer if we put on the profiles. So we've got dissolved iron here on the left and iron isotopes here. The first thing that pops out of the profiles, and these are the ones going west-east, is actually there's a big change in iron concentrations, even though you can't really see it necessarily with the colors. So as you go from west to east, you're losing about half of your dissolved iron. That's quite a large change. But if we look at the isotopes, there's actually very little fractionation. So we're seeing a big loss of dissolved iron from scavenging and particle interactions, but no isotope change, which for us is, I think, a big deal because, obviously, if you want to see a source signature transported, you have to show that it doesn't get attenuated over transport. And this is also something we've seen on the US cruise in the North Pacific, where the water masses pass through the particle veil. There's a lot of loss of dissolved iron, but there's no fractionation. So that was pretty exciting for us. And it's just emphasizing again the point that the source signature is persisting for 3,000 kilometers. So I think, at least in the North Pacific, this is a really nice example of those, sources that come from sediments in Japan are being transported right across the North Pacific, all the way to Alaska. Now, if you zoom in on these stations, there is a little bit of noise there. There is a bit of difference. But actually, what was nice from Hannah and Matthias's work is that, we're looking at this section straight across. Actually, the circulation doesn't really do that. It decouples from that section and heads off into the Gulf of Alaska at this is about 1,000 meters depth. And so if you sort of compare stations on the flow paths, you actually see even more agreement in the isotopes. So we can actually show that there isn't any fractionation between, say, this station and these ones right in the Gulf of Alaska. So what we're seeing is the persistence of the sediment plume and the primary isotope signature all the way into the sort of the armpit of Alaska, for want of a better word. So I think that's pretty exciting for us. Hannah will be writing that up soon. So, still much to be done on this topic, but I think we have some nice iron isotope signatures in sediments. We think they make them into the water column. Sort of put an asterisk because I think we're still working on that one. And we're seeing really long distance transport of this signature without any change, despite the iron loss. So I think it's quite promising. We're excited. One last thing I should say is that you might think, we went to Alaska and we measured iron isotopes and we got a number and we're tracing it back to Japan. How do we know it's not coming from the Alaskan margin? Well, actually, the really nice thing about geotraces, I've taken off the plot anyway. The really nice thing about geotraces, and you would see if I'd left the plot on, is that this plume doesn't have any radium. It doesn't have any radium-228, which means it's at least 30 years old. Radium-228 would come from the sediments alongside iron. So this plume isn't coming from the local margin, it's being transported and it's taking at least 30 years to do so. So it's really important to measure different traces. Don't just go out and measure iron, also measure radium and thorium and everything else that gives you all these different rates and fluxes. So that's me almost done. The other thing I wanted to say with the jigsaw is obviously this is work that's been done by a lot of people and with a lot of help. So I wasted a bunch of time yesterday putting all my lab and major collaborators into the puzzle. So thank you.
Okay, great. Thank you very much. My name is Yao Fu, as Dr. Hu has introduced me. So today I'd like to show you some latest results about the OSNAP observations. And as you'll see, these are my co-authors and collaborators of the OSNAP project. And OSNAP is fully supported by the National Science Foundation for the US component. but you will see during the talk that OSNAP is a huge collaboration program that involves different countries. Right, okay, so I suppose that everyone in this room is an expert of the AMOC, so I will try to be brief. So in general, we think that the AMOC consists of a northward flowing upper limb that's warm. It also consists a southward flowing lower limb, which is cold. And the water sinks in the subpolar North Atlantic, in the North Atlantic part. So in this setup, it can so the AMOC carries a huge amount of heat northward, but at the same time, because it's a water flowing, it carries also oxygen, carbon, nutrients, and different tracers. And therefore, it is very important for the global climate, but also for the ecosystem. So to understand such a huge circulation system, scientists around the world has get together and set up a observation program, which is called Overturning in the Subpolar North Atlantic program, OSNAP. So as you see, we have two sections in the subpolar region. So here is the Labrador, well, Canada and Labrador. And then in the middle is Greenland. We have Iceland here, and this is UK and Europe. And we have OSNAP West covering the Labrador Sea, and OSNAP East covering the eastern part of the subpolar region from Greenland to the UK, to Scotland. And along the section, we have a lot of moorings, roughly 60 moorings, and measures temperature, salinity, velocity, and different biogeochemical tracers. But because this is an international collaboration, I would like to acknowledge each country along the section. So at the very western end, we have 3 moorings operated by Canada. Although it's sub-awarded from NHUI money, it's NSF money, but it's operated by Canada. And then over the Labrador Shelf, continental shelf slope, there are about 10 moorings operated by a German group. And then on the western side of Greenland, it's the US mooring operated by a HUI group. And then on the eastern side of Greenland, we have 2 ARI groups, but both operated by a HUI and also University of Harvard. And then we also use 2 OI moorings in the center of the Erminger Sea. And on the western side of the Rickians Ridge, the moorings are operated by a Dutch group. And then on the eastern side of Rickians Ridge, it's moorings operated by University of Miami. And then in the Iceland Basin, we have 3 tall moorings, which is operated by the UK NOC. And at the very east end, in the Rockall Trough, we have 4 moorings operated by a UK SAMS group, which is a Scotland group. So as you see, this is a international collaboration, and we spent effort to understand the variability of the overturning in the subpolar region. And so typically, as I have already mentioned, this is basically a deployed log of a mooring. So along the mooring, we have different type of instruments. We have ADCP measures velocity for a certain range. And then we have a current meter that measures velocity at a fixed depth. We have macro-CDD measures temperature and salinity at a fixed depth. And then we have some biogeochemical sensors like oxygen. So this is just a figure showing you deployment of the moorings. And then by utilizing those instruments, we can produce a section of velocity, temperature, and salinity. And from there, we estimate the transport, basically the meridional overturning circulation, the meridional heat and freshwater transports. So this is just an example of a mean salinity section, but as I mentioned, we have also temperature and salinity. So these are the equations measuring, calculating the AMOC, meridional heat transport, MHT, and the meridional freshwater, MFT. So essentially, we calculate the A mark. First, we calculate the stream function, overturning stream function in the density space. As you see, we integrate velocity zonally in x direction, which is longitude. And we also integrate the velocity vertically in the density bins. So it's calculating density. And for MHT and MFT calculation is essentially similar, but involves temperature and salinity. So here is just an example about the overturning stream function. The thin lines represent the overturning stream function each month, and the thick lines are just the mean of the overturning stream function. And now by picking up the maximum of each monthly overturning stream function, which is indicated by those open circles, we find the overturning strength. Because essentially, as you remember from the very beginning, so the A mark has a northward flowing upper limb and a southward flowing lower limb. So the strength is essentially measured by the maximum of the stream function, which represents essentially the northward, the total northward transport in the upper limb. And also, we name the density of this maximum overturning as the sigma mark. Sigma is essentially potential density, and sigma A mark or sigma mark is just the density where we find the maximum of the overturning. All right. So by placing those dots in a sequence, I can show you now the overturning time series for the full section, which is in black across the whole subpolar North Atlantic, and also the overturning across OSNAP East and OSNAP West. So as you see, the 8-year data now, although we have 10-year data calculated, but still in the preliminary phase. But the 8-year data shows a very consistent results from compared to the very first 2 years of data, which is the eastern part of the subpolar overturning contributes dominates the overall mean and variability of the subpolar overturning. And the western part, which is the Larboard Sea, only contributes a smaller portion. But of course, we see that across the whole section, OSNAP East and OSNAP West, there is strong variability. So I picked the title of this talk as Interannual variability. So the way that we investigate interannual variability is first by determining seasonal cycle, right? So when we have the seasonal cycle, then we can simply subtract the seasonal cycle from the time series and derive a mark time series without seasonality. And then by superimpose a low pass filter time series, now we can see clearly the interannual variability of the A mark across OSNAP East and OSNAP West. Okay? But of course, we want to understand what is driving the overturning variability on interannual timescales in the subpolar region. And the way that we do it is, okay, so it works. The way we do it is by performing a so-called lower limb volume budget analysis. So you could think about the volume budget as a water tank, and there's well which add water to the tank, and there's a sink that reduce the water from the tank. So the idea is basically we want to focus on the lower limb, which is defined as the volume below the sigma mark, as I just mentioned from the overturning stream function. So here is just a simple equation about the volume budget. There are 4 terms. So the first term is just the change of the volume in the lower limb, which is derived by integrating the depth from the sigma mark isopignal to the ocean bottom. We know the bottom doesn't change in this time frame, but the isopignal surface changes over time. So basically, we are measuring the vertical displacement of a certain density surface. And then the next term, which is called surface force water mass transformation, essentially that's a measure of how much water is converted from the upper limb to the lower limb by surface buoyancy forcing. For example, cooling at the surface, the water becomes denser and then sinks and then becomes a water in the lower limb. So we can think about transformation as the mechanism adding water to the water tank. And then the third term is export, but it's equivalent to the A mark, especially for OSNAP West, because essentially for the lower limb, the opening of the labyrinth is the only pathway for water leaving the basin. So so basically export is equivalent to the overturning across OSNAP West. And this is a mechanism reducing the water from the water tank. And then, but for the OSNAP East, the export term is slightly different because we have two boundaries. One is the OSNAP East section, but the other boundary is called the Greenland-Scotland Ridge. And over there, we have measurements of the overflow water crossing the GSR. So by differentiating the two, we get a net transport that's leaving the basin. But because the variability of the overflow water across GSR is relatively small, so essentially the variability is determined by the overturning circulation across OSNAP East. So the last term is called residue. We do not calculate residue explicitly, but rather as just a difference of the first three terms. Because as you see here, I listed all the data set we use to calculate the first three terms. The first one is just calculated from density. The second one is a combination of surface fluxes like heat flux and freshwater flux and density. But the third term is essentially OSNAP observation and GSR observations. So these three we can calculate independently, but the last one is a residue. And the residue typically measures or includes the physical processes that we don't really resolve. For example, dipycnol mixing, or any measurement error or uncertainty in this calculation. All right, so I will show you the results, but don't be intimidated by the plot, the lines. I will walk you through one by one. So here, the equation is here, essentially as a reminder. And then please note that all the lines shown in this plot has been deseasonalized, and then after a 1-year low-pass filter, so that it represents interannual variability. And the green line represents the volume change, essentially the measurement of this vertical displacement of the isopycnol. And then the red line represents transformation, adding water to the lower limb. And the blue line represents export, which is reducing the lower limb volume. And now, because it's a very simple budget analysis, it's a water tank. You can't do wrong with it. Essentially, what we can do to determine the relationships between these variables as simple as a lead-lag correlation. But before showing you any lead-lag correlation analysis, I just would like to mention the residue. So as I mentioned earlier, residue measures the processes that we don't resolve, like mixing or any error. However, if you remember at the beginning that I showed that the overturning circulation across OSNAP East section is around 16 swerdrops. But now, as you see, the horizontal dashed line represents time mean residue, which is about 2 swerdrops. I think it's quite remarkable that using those independent observation data, we can close the budget in the mean by roughly 2 swerdrops. But anyway, it's not the case for OSNAP West or the Western Basin, because overall, the overturning circulation strength across the labyrinth is only swerdrops, but 2 swerdrop residue is quite high. But anyway, let's continue with the lead-lag correlation. Again, I'd like to emphasize transformation is the term adding water to the lower limb. And so we think that whenever a transformation event occurs, we should see that the lower limb volume increase. And that's what the lead-lag correlation shows. So at 0 lag, we see the maximum positive correlation, which basically means that whenever a high anomaly of transformation, there's an immediate response of the lower limb, which is increase. And then it's the case, a similar case for both OSNAP East and OSNAP West. So next, I'd like to examine a different pair of variables, which is between transformation and export. So the relationship we found is essentially that the two are maximally anti-correlated when transformation leads the export by a few months. So that basically means that at this point, I think it's safe to assume that whenever transformation occurs, the lower limb volume increases, and a few months later, we see an increased export of water. Okay. However, although both sections shows a similar story, but when we examine the next pair of variables, which is between volume change and export, we only see something reasonable in the eastern side, but nothing significant on the western side. As you see that here, volume change also maximally anti-correlated with export when volume change leads the export by a few months. But the correlation at OSNAP West, or the Western Basin, is really low. It's not significant. So basically that means at this point, we can only confirm the relationship exists in the Eastern Basin, but not in the Western Basin. All right, so I will briefly summarize about this part. So by using an 8-year time series, we can now investigate interannual variability of subpolar overturning. And through a framework of lower limb volume budget, we found the expected relationship between the three variables, which is transformation, volume change, and AMARC. But essentially, we want to build a relationship from transformation to AMARC because we know transformation adds the water and export is the response. And but then we can only confirm such a relationship in OSNAP in the eastern subpolar basin, which is enhanced transformation leads to increased volume and then leads to an increased export. But the relationship in the western basin is not clear. But of course, the reason for this, there might be, well, we don't exactly know why the relationship in the western basin is not clear. But one of the assumptions is really that because the signal to noise ratio in the western basin is so small, the overturning is just swerdrops, but overturning in the eastern basin is 16 swerdrops. So the signal to noise ratio still in the observations we have a lot of uncertainty. But next step is really want to understand what is driving, what else is driving the interannual variability of subpolar AMARC, like what is the role of the wind, and also as we talk about freshening, because we have Arctic sea ice melting and also land ice melting, and also there's a subpolar system internal freshening going on in the subpolar region. We want to understand what is the role of freshwater anomaly on AMARC variability. And next, I'd like to show you some results beyond AMARC observation, right? So the OSNAP program is established, designed to measure the AMARC, but we have been trying to explore more beyond the AMARC observation. So here on the upper, upper right now, my right now. So it's a snapshot of velocity section, which is I produce. But then on the left side, it is a anthropogenic carbon concentration across the OSNAP section. We do not measure carbon directly from the OSNAP observation, but we can derive a anthropogenic carbon concentration from a salinity budget analysis. And then by combining the two, we have a section of anthropogenic carbon transport. And then by integrating the transport across the whole section, we derive a time series for anthropogenic carbon. all these three plots are done by Claire. And what we see is that for across the whole section and OSNAP East, we have a northward transport of anthropogenic carbon. But what's surprising is really that across OSNAP West, which is across the Labrador section, we have a southward export of anthropogenic carbon, which means that the Labrador Basin is exporting anthropogenic carbon to the North Atlantic. So that's one example about anthropogenic carbon transport. Another example is really about oxygen transport. So along the OSNAP lines, especially across the western boundary part of the eastern subpolar section, and both boundaries across the Labrador Sea, the OSNAP moorings are equipped with oxygen sensors. And by utilizing ship sections and also biogeochemical algos, we can analyze the property change of water masses along the pathway of the waters. So here is well, TS diagram, but color coded with oxygen. So when we start from the upper corner, so that's water masses across the East Grand Current region, which is this part, east of Greenland. And we see that the water is relatively warm and relatively saline, but also low in oxygen. When we progress, around the southern tip of Greenland, we see that the water masses become slightly cooler, slightly fresher, but also slightly richer in oxygen. And when the water masses goes around the Labrador Basin and leaves Labrador Basin, now we see the water masses are strongly cooled and freshened, but now very rich in oxygen. So to summarize that, we use a schematic that shows the process of ventilation. So the oxygen was low at the beginning when it goes across the OSNAP East part and then go around Greenland and becomes slightly rich and in oxygen. And when it goes around the basin, because of convection and mixing, the water becomes very high in oxygen. And so from there, this is the very first oxygen transport across OSNAP West section that has ever been produced. And so basically, these two examples demonstrate that the OSNAP observation is not just a physical observation, but also can serve as an infrastructure and tool for understanding biogeochemical processes. All right, so that's the last slide. Thank you very much.
We had some really good talks this morning that, were your really nice question, data, answer, conclusion. You're not going to get that for me. This is more of a programmatic overview of the shelf program, which at its core, we want to better understand spawning dynamics of fishes in the Eastern Gulf of Mexico. And we do that by DNA barcoding fish eggs that we collect and that others collect. And there's sort of two main take-homes that I want you guys to have at the end of this presentation. One is that we're coming up with a lot of different data products to address a number of different questions, from basic questions as simple as what species are spawning, when, and where, right? We know surprisingly little about that globally. But then these data can also be used to address applied needs, such as feeding into stock assessments or understanding broader ecosystem change or stability within the Eastern Gulf. Okay, so that's one part that I want you to take home. The other part is that we're open for collaboration. In fact, you'll see here, is Yonggang in here? Hey, Yonggang. So we've collaborated with Yonggang. We're currently collaborating with Weiyi. He went on a recent cruise, and his student is going on the next cruise along with some of his colleagues at Texas A&M. We're collaborating with David Nahr, who's not here, and his students on bathymetry work. And because this is a long-term program, even if you don't have any ideas today, the door is open, right? So if something emerges and you want to come talk to us, we welcome that. I want to recognize the co-PIs on this, Maya, Steve, and Ernst. Yes, Ernst is still involved. I just talked to him 2 days ago. Yeah, it was good talking to him. By the way, Ernst's compound is amazing. It is cool. All right. how did you magically get this thing to, there we go. So Shelf began in response to RFP2 from the Florida Restore Act Centers of Excellence program. And their central goal was to have a program that built a comprehensive ecosystem monitoring program in the Eastern Gulf. It says Gulf of Mexico, but we're really relegated to the Eastern Gulf. And I'm going to use this slide as an anchor to describe the work that we've completed so far, as well as the ongoing and planned work. And so just briefly, this is what this looks like. It began in 2017 as a 2-year pilot, a rather ambitious 2-year pilot, I might add. And we're going to dive into each of these. I'm just giving you the timeline to start. The second phase of Shelf is when our egg monitoring program actually began. At the beginning of that, we also further delved into different ways of barcoding the eggs. You know, as we're trying to build this program, we wanted to do it right. We're currently in the third phase of Shelf. The egg monitoring program is ongoing. It's a pretty well-oiled machine at this point. And then we have the arrival of some targeted studies. And this is a really cool part of how the Shelf funding is structured. And I'll get into that in a bit. And then last, I will just barely touch on the future of Shelf, the next 3 phases, 4 through 6. But we will continue to build this long-term egg monitoring time series, as well as those targeted studies. All right, so as I mentioned, it began with a pilot. And that pilot actually dovetailed off of some successes from RFP1 with some efforts led by Mackenzie. Where's Mackenzie? She's not in here? She wasn't here. Yeah, she left for this one, fine. I understand. She's actually seen it. So Mackenzie, Maya, Steve, and Ernst showed that they could successfully DNA barcode fish eggs, right? And so based on these successes, we developed this pilot, and we did several things. One thing is that we applied what's called the daily egg production method to estimate spawning stock biomass. I see the glazing of eyes. Talking about stock assessment. I'm not going to go into it, but just know that traditional stock assessment does hindcasting. It looks at the last 3 to 5 years and determines how that population was doing. The daily egg production method puts your estimates of the stock in real time. And we applied this to vermilion snapper, and lo and behold, our estimates were consistent with those provided by traditional stock assessment. So we view that as a success story. That work is in prep. Notice I don't identify who's in prep. We're kind of in a not it mode right now, I think. Now, because we were collecting eggs all over the western, at least the northern West Florida shelf, we were able to also just simply map out the spatial dynamics in fish egg densities. And what we found is they tended to be more abundant near shore with this apparent hotspot near Cape San Blas. So then Yang was able to explain why that hotspot may be using physical oceanography models. And then looking forward, we estimated egg retention and export dynamics and found that those spawned on the inner and middle sections of the shelf tended to be retained, whereas those spawned on the outer section of the shelf tended to be exported out of the system. And this was part of, a lot of you guys weren't here when V. Wen was doing her master's work, and we published that work last year. During this phase, we also explored a lot of other different tools and methodologies. Stable isotope analysis, we used the CBAS, some acoustics, etc. These ultimately haven't been incorporated into the second and third phases of shelf. But again, the door is always wide open because we do have that flexibility as we come up with these targeted studies. Okay, so, we threw everything, including the kitchen sink, at that pilot. And we submitted our final report to the program management team, and they asked that we prioritize the egg monitoring component of the program, but then with that flexibility and funding for targeted studies. These targeted studies can be used to address interesting patterns as they emerge. So, diving in more deeply and better detail on things. We've also used them to pivot when circumstances arise that don't allow us to do our original plan, right? And I'll give you some examples of both of those in a minute. But those are really nice levels of flexibility in how the funding is structured. Don't always get that. Okay, so at the conclusion of the pilot, we then moved into the second phase where we were set to begin the egg monitoring program. And so we collected eggs in 2019 across 49 stations in, the northern and mid sections of the shelf. We processed them, and the barcoding was completed by early 2020. Now, up to this point, we've been using individual egg barcoding. So that means you meticulously pick each egg from a plankton sample, and then you process each one of those eggs individually. We wanted to know if we could increase our throughput and reduce our costs by doing what's called metabarcoding, which is where you barcode a sample in composite, so all of your eggs from one sample together. We did that from over 4,700 eggs. We recovered 37 taxa, and the taxa that we recovered seemed to be the ones that we would expect to find in the places that we found them, right? So it made sense. It looked like the metabarcoding approach may have been working pretty well. But we still wanted to compare the metabarcoding versus individual egg barcoding. And so we can do that here with the pros and cons. And where metabarcoding really shines is the cost. It's an order of magnitude less than individual egg barcoding. And this doesn't even factor in the human resource costs. This is just purely analytical costs. But that's the only place where metabarcoding wins. And the other parts are actually way more important. So metabarcoding is only semi-quantitative. You only get presence absence. You can't get densities, and so you can't create indices of abundance or these kinds of things, which are needed for the stock assessment stuff that I mentioned earlier. Whereas individual egg barcoding is quantitative. When you barcode eggs as in a composite, they're destroyed. You can't go back to them afterwards. And so if something happens where you want to go back to that sample, you can do that with individual egg barcoding, but not metabarcoding. And then finally, and this is a huge one, we had a ton of false positives and false negatives with metabarcoding. And so the data quality just weren't as high. For example, we would sometimes get more taxa back than we had eggs, which of course is impossible. Whereas with individual egg barcoding, we have very low prevalence of false positives and negatives. So we decided moving forward, the best way to have the best quality data, even though it costs more, even though it's more time consuming, was to use the individual egg barcoding. Okay, so, we're now 3 years into this thing, right? We got the 2-year pilot, we got the, we're figuring out this barcoding stuff. We're all ready to go, ready to start the program and collect eggs in 2020. This is all I have for this slide, because what else are you going to put up here? Somebody coughing? So we didn't feel comfortable sending, we were going to go out in summer of 2020 using the Weatherbird, and we didn't feel comfortable asking students and research staff to work and live in those tight quarters. Fortunately, this is where Shelf takes a little pivot that ends up being a positive, because we know that the CMAP program, which is largely run by NOAA, had been collecting eggs on the West Florida shelf, as well as the rest of the U.S. continental shelf in the Gulf, for about 10 years at this point. But they were just archiving the eggs. They didn't have anything to do with them. And so we reached out and we said, hey, we can DNA barcode those eggs, pass along the data back to you, and they said, yes, that would be great. So it was a win for NOAA. It was a win for Shelf. They collect their eggs using what's called a continuous underway fish egg sampler instead of a plankton net. It gives you very clean samples because it selects for egg-sized particles. This is where the selection process happens. We call it the magic margarita machine, because it looks like something you might see on Duval Street or Bourbon Street. And so they gave us their samples, the ones that they had available, which ended up being 2013, 2014, and 2019. So in this second phase of Shelf, we barcoded those years, 251 stations, over 160 taxa were identified, and we had about an 80% barcoding success rate, which is good. It's on par with other programs. So this work during the second phase was largely done by Keith Keel for his master's thesis, which ironically, when I left here, I ran into him as he left FWRI for lunch. He's employed there. But I'm just giving you high-level results with his data for those 3 years. He found some structuring in the fish assemblages along an inner to outer shelf gradient, and among other things, and he published that work earlier this year. And so this is just a very cherry-picked example of some species that we expect to see in those inner, middle, and outer portions of the shelf. And so sure enough, where we found their eggs corresponded with what we think we know about them, right? So that makes sense. Another exciting finding from the second phase of shelf is we identified some yellow wedge grouper eggs in the vicinity of Pulley Ridge habitat area of particular concern. And this is a deep water grouper. It's a very data poor species. It's managed. And so, knowing something about where they're reproducing is really important. And this habitat area of particular concern was established in part to help support spawning of commercially important species. So it seems to be doing its job. We'll come back to this in a minute. Okay, so that led us into the third phase of shelf, which is where we currently are. And so we're continuing our long-term egg monitoring time series, working with CMAP still. Even though we're starting to go out on our own cruises, we're still using those CMAP eggs. So that's a great collaboration. These first 3 years here are from the second phase that Keith worked on. And in the current phase of shelf, Alexis Mitchum is barcoding these eggs for her dissertation work. And so 2022 went off well. 2023, the COOFs did not work. The CMAP COOFs did not work. It was inoperational. So I'll show you in a minute what we did since we didn't have samples for 23. 2024, they were able to sample the northern part of the West Florida shelf, but then Helene chased them out. They got down to about Tampa Bay area. And then they are now scheduled to go out actually, I think the cruise is ongoing right now for the 25 collections. Now, this cruise was originally canceled due to federal funding cuts. And so we did not think it was going to occur. We have an amount of allocated ship time, and so we reallocated it, according to our plans. And so we're also going to be going out next week. Our cruise pushes up on Wednesday. So this is kind of an interesting opportunity to have somewhat synoptic sampling between the NOAA vessel and the FIO vessel. Okay, so what do we do about that 2023 money and time that we had budgeted? Well, when we barcode these eggs, we have to choose which ones to barcode. I've got some R code that I write that selects the samples randomly. But we leave a lot of samples that aren't barcoded. And so what we did with those funds is we looked at all of the archived samples in the Pulley Ridge area and barcoded them with those funds. And sure enough, we found more yellow edge grouper eggs, as well as other species that are pretty data poor, like Cubera snapper. So this is that flexibility that I mentioned earlier, where we're able to pivot to address something both as it emerged and in response to a logistical challenge. Okay, so I will go over the targeted studies that we're running during this third phase. The first is to explore the seasonal dynamics in spawning on the West Florida shelf. So I'm going to dive into each of these. I'm just introducing them for now. The second is we're asking whether we can link adult fish abundances to egg production. And so these first two targeted studies are to better understand spawning dynamics on the shelf. And then the third question was, we wanted to know if our collections, which are made 3 meters below the surface, there's an intake pipe along the ship 3 meters below the surface, whether that's truly characterizing eggs throughout the water column. And then last, we wanted to know if we could improve our DNA barcoding success. Again, we'd had about 80% success. That means we're leaving about 20% on the table. We wanted to know why and can we improve that? And so these questions were to explore methods and assumptions. All right. You can't read this. That's fine. There's no way you can read that. Maybe you guys. So this is a spawning table. Each row is a different species of managed fish. And these columns to the right are months of the year, January through December. And so if a cell is color coded yellow, that's not yellow, that's gray. If it's colored gray, that means there is support that they're spawning for that species during that time of year. If it's black, that means that is peak spawning. So for example, the top row is the king mackerel. It spawns from May through October, with peak spawning in June through September. The gray highlighted columns here reflects when CMAP samples. And they chose that time of year because a number of different mackerels, groupers, and especially the snappers, and especially most importantly, red snapper, that's the time of year that they spawn. That's why they chose that time of year. And that's great. It does capture a number of species, but it also leaves a lot of other species that will not be detected by only sampling that time of year. And so for 2 years, we are supplementing the CMAP sampling with our own sampling in March, June, and December, representing the three additional quarters of the year. And you can see that does pick up nearly all of the species on this table. And we're doing that using our own COOFs. So we built the COOFs in the shop. We built it to spec with the CMAP COOFs. In fact, we brought the CMAP COOFs back here to USF. And this was one of Jimmy and Guy's last projects. Garrett Muller, for those of you that remember him, was very much a part of this. And then last June of 24, we had our inaugural cruise. Here's our crew members. There's a couple of them here. And it was a success. So this is what that first cruise track looked like. So this is also important to consider if you guys want to collaborate with us in some way. Now that cruise track isn't always like that. You know, we sail according to a lot of things, including surface conditions. So we try to make the cruise as comfortable as possible. So we don't have to retrace that particular track exactly. And these are the cruises that we performed so far. And again, we have another one that's leaving next week. The second targeted study, again, is whether we can link adult fish abundances to egg production. We're doing that by combining two different data sets. So my lab has been running a reef fish survey program on paired artificial and natural reefs off the coast of the Tampa Bay area since 2013. So we go out and we do diving surveys on these reefs quarterly. We're always also looking for divers. So it's another plug here. And so we've got some good long-term dynamics of the reef fish abundances and densities on these reefs. And so what we're doing is when we go out for these quarterly full shelf surveys, we also spend 30 minutes mowing the lawn over top of these reefs, collecting fish eggs. Again, I think that's some work that Alexis is going to be taking over. She's also been doing some eDNA samples from above those reefs as well. And so we have a combination of visual survey data, eDNA, and eggs, which is kind of a cool combination. And then these are the collections we made so far. Next, with our two studies about assumptions, again, we're sampling in the very upper layer of the water, and we do that because fish eggs are buoyant, and so they should ultimately make their way to the surface. But we wanted to know if our sampling did, in fact, collect those eggs, or characterize the entire water column. And again, we went back with some archived samples from CMAP where they were using mock nest sampling. And this is some work that was led by Nina while she was an undergrad, and she's now a grad student with us. And the answer is kind of, for some species, yeah, surface sampling is in fact characterizing them. And for other species, maybe not. And that's fine. It just contextualizes our results. This work is currently in review. And then last, with our assumptions work, Alexis led these efforts to determine why we were having barcoding failure and can we improve it. I'm not going to go through all this, but this is kind of the flow chart of what she worked on. And the ultimate answer to this is, yeah, she improved it by 7%, something like that. 7%, but it's a doubling of cost and it's a doubling of effort. So it just wasn't really worth it. So unless we have a really good reason to go back to our samples, we probably won't do that moving forward. Okay, so we checked out these two. I don't know if that's a publishable paper. You can think about that. Hopefully this is a publishable paper. And then we're still collecting eggs for these top two questions. And Maggie said she would come back and talk next year, but I'm not gonna say I'll do the same. I'll come back in 3 years. Next time Sean Ben twists my arm. Oh, by the way, we were saying how new we were here. This is my 15th year, so I get some kind of service gift, like a paperweight or something really nice, I'm sure. You know, in addition to our papers, we've become more active at disseminating our products to different conferences. Seems like we go to New Orleans a lot for some reason. But beyond these sort of more typical science conferences, we also, Maya and I presented at in front of the Fishery Management Council about 1.5 years or 2 years ago. It was very well received, very positive, and they seem to be extremely excited about the products that we can potentially provide them for the sake of not only stock assessment, but broader ecosystem change. And we've also become more involved in outreach. This is an activity that Maya's group did at the St. Pete Science Festival using this really cool paper that Mackenzie and Maya wrote on DNA detectives and how to use these kinds of activities to get especially elementary school age kids interested in this kind of stuff. And then I was contacted by the Canterbury School here in St. Pete. They had received about $1,000 to build a drifter. That picture didn't show up. To build a drifter, it's got, well, it had solar power on it and satellite pinging capabilities. And they really wanted it to catch the Gulf Stream. And so I worked with Young Gang, and we were on a cruise out here mowing the lawn. And I said, where should I drop this thing? And we dropped it. You can't see it. We dropped it on March the 14th of this year, and it continued to ping until just a couple of weeks ago, just before Hurricane Aaron. So I seriously doubt it's going to come back on board after it. But it was really cool to watch this thing on a daily basis. I probably bugged Young Gang more than he wanted me to. But it was just kind of meandering along in here, got stuck in a couple of eddies. It actually moved between the keys and the Tortugas back onto the shelf. And I was like, oh no, this thing's never going to catch it. But when it caught it, it went from doing this for months to shoo, it went up and yeah, got off to, the coast of sort of the northern Chesapeake Bay area. So that was really cool. All right, I'm out of time, but looking forward, so the way this funding works is we apply for renewals every 3 years or so. It's non-competitive. And if we are funded for the duration that we're eligible, it'll last through 2035, possibly longer if we have some no-cost extensions kind of embedded in. So that should cover nearly a quarter of a century to build this time series. And that'll help us to begin to understand the spawning dynamics on the shelf, as well as how species may respond to both disturbances as well as things like rebuilding plans that management puts in place. And it's my vision that beyond the funding from Florida Race Up, either I hope that we can leverage some kind of funding once we show how important these kinds of programs are, or we pass the program along to somebody like NOAA who can continue it for themselves. And then again, we have that flexibility in there that we can respond to issues as they emerge, and we can incorporate all kinds of complementary tools. And so again, that's just to bring the whole thing full circle. That's where you guys come in. we keep an open door. If you guys want to collaborate on anything that either has a direct or even an indirect effect and connection to shelf, we'll certainly listen. Thank you.
So we're gonna go off to the Mediterranean. Thanks to a few years ago, I had a scientist from Alexandria University came and worked in my lab for about 6 months, and we only got about 6 papers out of his 6 months there. But and I was on Olga's dissertation committee in like the early teens, I think, or something like that, and and worked on a series, or she worked on a series of samples that kind of disproved or the whole story about, whether this thing is invasive or not is pretty controversial. Okay, what did he said up and down, but. Yeah, there we go. So what I want to do is, for those of you who have no idea what I'm talking about, larger benthic foraminifera. If you go to a store that sells limestones and you see some really pretty limestone, fossiliferous limestone slabs especially from Tunisia and some of the other places along the Mediterranean region, they're probably full of larger benthic forams. And if you are particularly interested, you could come by and see some in my office. But anyway, actually, Amphistegina lobifera is the genus, or one is from the genus, and one of the species that I worked with for my master's degree a few years ago at the University of Hawaii. And so what we're talking about, what I'm going to do is tell you a little bit about why I worked on it in the first place, and then talk a little bit about the Mediterranean and some possible consequences of climate change. So larger benthic forams, as I said, they look at some incredible insections or slabs at Home Depot or whatever. The larger benthic forams are morphologically defined by their large sizes and complex interiors. And those complex interiors, at least in modern species, and we suspect in the fossil species, are associated with housing of algal endosymbionts. And they range, because they've been around longer, or actually, yeah, larger benthic forams have been around longer than symbiont-bearing corals, or algal symbiont-bearing corals. Only a few of them have symbiodiniaceas. And some of the older species actually have red algal symbionts. Most of the ones that really became important in the Paleogene, the Cenozoic, have diatom symbionts. Again, some of the older taxa have chlorophytes, and then another group has dinoflagellates, very much related to some of the coral taxa, what are found in some of the coral taxa. And the other thing is that these that look like, they're called actually porcelaneous taxa, and you can see that they have a diversity of kinds of symbionts, including some that aren't shown here that have diatom symbionts. They precipitate high mag calcite, and so their chemistry, their shell chemistry, is somewhat different than the rotolids, some of which have high mag calcite, but many of which have what we call intermediate mag calcite. That's maybe 4%, but not not up to 10 or 15% magnesium. All righty. So, and this is just, I mentioned Tunisia earlier, and I got to go on a field trip many years ago to Tunisia, and it was one of the most spectacular field trips from a lot of points of view. First of all, there wasn't a lot of vegetation covering the beautiful rocks, and again, lots of nice forams. All these plates are a type of actually deeper dwelling, low light, oligophotic light livers, the things that live in the lower light, not. Okay, so the importance of carbonates to humans, they've been kind of important to us as humans for a long time. They shelter in building stones, and those of you from Florida, we get a lot of water, our water from the Florida aquifers. And so then they're also recognized as major reservoirs for fossil fuels. And what I was doing earlier this week was attending the American Association of Petroleum Geologists image meeting, but I was giving a unfortunately a memorial talk for the person that I really got to know on that Tunisia field trip. That and we wrote, I don't know, several papers after that. So yeah, carbonates are kind of important, have been kind of important to people for a long time. Amphistegina lobifera, it's not all that large by larger form definitions, but it's robust. It has diatoms, symbionts, and it's a modern species that thrives in clear, nutrient-poor coastal waters throughout. And this was a paper from 2000 that basically mapped the genus in the throughout the Indo-Pacific. Lobifera does not occur naturally in the Caribbean Atlantic province. Okay, so it wasn't actually formally described until 1976, and earlier records were just identified as species or as one of the other more common. Well, Lawsoni is the most common modern species. And there's a German scientist who claims that she's barcoded, I guess, something that can still be called Madagascariensis. But anyway, so, but in drilling and the Funafuti Atoll back in 1900, they found that the major constituents of the sediments, not the limestones necessarily, but the sediments associated with Funafuti Atoll were Amphistegina shells. And Amphistegina and Calcarina shells in Holocene reef sediments have been recognized, especially the U.S. Geological Survey professional papers from post-World War II up through the 60s and 70s. And one of the things that got me into forams was that, first of all, I was a girl pre-Title IX, and so forams were a good, safe thing for girls to work on. Little did they know that I was going to turn that into carbonate research, but whatever. But at the time then, about a quarter of Hawaii's beach sand were foram shells, and because it lives in the shallowest water and it's the most robust and sturdy, the majority were lobiferous. Okay, so again, lobifera is shallow dwelling, highly adapted to clear, warm waters, enhanced by hydrodynamic sorting, and because it's relatively robust, it's enhanced resistance to abrasion compared to a lot of other carbonate components, because I did some comparative studies actually in my graduate work, in which I compared the production by lobifera to the standing crop, if you will, in the sediments, and it was 10% versus 25%. So they were producing about 10%, but responsible for, in the beach sands, about a quarter. Okay, so, but we've got some hints from evolutionary history as well. And that is that this genus was documented in the early Eocene. And so it evolved under higher PCO2, as did the ancestors of the diatom symbionts. And the people have done, because lobifera and lawsoni are so easy to culture, people have done a lot of experiments with ocean acidification with them, and the results were all over the place. And it turns out the results are all over the place because it depends on what ppm, what the concentration of CO2 they worked with. If they worked with over 1,000, they got dissolution. If they worked with under 1,000, they got enhancement. And that's about right for their evolutionary history. So again, they were the genus, not the species, but the Amphistegina were prolific around the Mediterranean until the Messinian salinity crisis, which was when the Mediterranean was closed off from the Atlantic, and the Tethys Seaway was closed, and so there are major evaporite sequences in the Mediterranean. There are records of Amphistegina in the Eastern Mediterranean from both the Pliocene and Pleistocene, but the current taxa, the current species, the genetic work has shown that they're directly descendant from those that have come through the Suez Canal since the opening of the Suez Canal, and they've proliferated in the Eastern Mediterranean. They're in the it's about, there's dozens of papers on how these dangerous invasives have altered the ecosystems and driven out other species and so forth. But I invite you to consider another possibility, and that is, before it was even formally described, there were reports of it even in the central Mediterranean, and it was described, it's been studied quite extensively since descriptions. Okay, and again, there's been this explosion of papers since about 2000, and this figure shows where, actually, guess I don't have it. It's down there somewhere. This figure is from a Tunisian graduate student of a late colleague from Italy. And so the thing is that even though the Mediterranean, if anybody's looked at it on the map, it's kind of not that low a latitude. and yet it has vast areas of clear, low nutrient waters, limited fluvial input. The only major river is the Nile in the Eastern Mediterranean. So the salinity, alkalinity, everything is pretty high. And I've got some maps to show some of that. Again, the Nile being and this is ocean color. I'm sorry, not ocean color. This is water transparency. And you can see that the nice blue water through a whole lot of the Mediterranean. Again, by the tourists like the Mediterranean. Elevated salinity. Look at that salinity in the Eastern Mediterranean. That's 39 and higher. And most people think of ocean acidification and pH. Think alkalinity. And alkalinity is, first of all, it's somewhat inverse to pH. And second of all, it increases, it's directly related to salinity. And although these forams are well, here's the total alkalinity, and then look at it's 2,600 versus what's coming in from the Atlantic is about 2,400 micromoles per kilogram. And these aren't even the new figures. And I started to say that although it's calcite, it produces calcite. Aragonite saturation is pertinent to the story, and look how high the aragonite saturation is across the Eastern Mediterranean into the Central Mediterranean. And sea surface temperature is pretty warm. This is October, so that's a nice warm 25 degrees. And yes, there are these pockets of cold areas to the north, but they also tend to be lower salinity as well. So we won't worry about the 4Ms don't go. I mean, other 4Ms go up there. Okay, so sea surface warming. Again, there has been warming in the Eastern Mediterranean. And so again, the advantages of warming allows these symbiont bearing really tropical, subtropical species to expand northward. The warmer it is, the greater the advantage of algal symbiosis up to about 30 degrees. And the increased advantages of calcification as a source of CO2 for photosynthesis. And actually, that was a topic that I was talking about in Houston earlier this week. Okay, so is lobifera an alien species? Well, the Commission of European Communities said that an alien species refers to a species introduced outside its natural past or present distribution. So it depends on how far past you go. But anyway, Langer originally said it was no, because they'd been there. Lots of reports, both fossil and even into the Pleistocene. But yeah, so Merck had found them from Turkey in several papers. But based on the European community's definition, it's no, it's not an alien. It's an alien species now in the Mediterranean, but probably not an invasive. What have I done? But at the same time, there's people who get really excited about invasive species. It's one of the 100 worst invasive species in the Mediterranean, and it's partly because they're producing all these sediments in the Eastern Mediterranean. But the question is dominance of one species and a count set. So what they do is they count 300 4Ms in a sample, and if Amphistegina makes up 250, it's only eliminating the other species if you count and do the proportions of the other 50 individuals, or a bigger count, because it's not taking the habitats of the species. And in fact, that was what various people have done. Is the group from Turkey that I've corresponded with over the years said, yeah, they're present in most samples, sometimes very abundant, but there's still a rich assemblage of the normal Mediterranean species. Olga, in her doctoral dissertation, showed that the smaller heterotrophic species dominated where there was a human impact and more nutrients and lower salinity or whatever. And Ahmed's work in the coastal areas of Egypt said, yeah, they're present, especially associated with coral algae, but in the Nile Delta region, they are not. They are never dominant. So, but they are altering shorelines, And so they're extremely abundant in sediment samples collected from the southwestern coast of Antalya, Turkey. Their shells accumulate as sand on the seafloor or carried to the shore by waves. They're deposited in small bays, changing gravelly shores to sandy beaches. And they're, in some cases, they're approaching, or they're over a half a meter deep sediment deposits. And the other thing is what they're doing is that most of the rocks that are eroding in this area are either old limestones that tend to degrade to lime muds or finer lime carbonates, muds and silts, or volcanics, which also degrade to fines. And so what they're doing is they're providing an input of sand-sized sediment so that the barnacles and bivalves and other things are actually preserved and helping build the beaches as well. So there's this is courtesy of Olga from Greece, and yeah, here's the forams. Here's some coral and algae. There's you can find pieces of mollusks, and so that's my point is that they're helping the other things not get broken down and washed away, as well as producing the carbonates themselves. And so is the return of lobifera perhaps ultimately beneficial? The size and density of the shells, again, make excellent beach sands. The relatively high salinity and alkalinity and warming provide ideal conditions for the shallow water, this very shallow water taxon. And they're producing beach sands. And if you go to the Mediterranean, a lot of this rocks that you find outside of any deltaic areas are going to be, again, carbonate rocks. Okay, so, and here's some of those beaches in Greece and Turkey that in the case of the Turkish beaches, the Russian tourists are apparently flocking from some of the things that I've heard and read that to the carbonate beach or to the foram beaches of Turkey, and lots of other people are flocking to from Northern Europe and even from the United States to the beaches in Greece and the outer islands. So that's all.
Multiple faculty in the College of Marine Science, plus a number of CMS students, faculty at USF St. Pete, particularly Heather O'Leary and her colleagues, faculty at Eckerd College and their students, and faculty at the University of South Carolina and one student there. Happy to say that not only are we supported by NOAA, who's the flagship here, but we also got some money from Bass Pro Shops. So they probably have more money than NOAA has.
All right. It's all right. No. Exactly. Should be able to. Yes.
All right. So brief overview. What are contaminants of emerging concern? What is the Tampa Bay Surveillance Project? Our objectives, a little bit of the programmatic components, and there's a lot of moving parts here. Wanted to talk a little bit about integrating all the parts and some kind of systems analysis and the modeling work, particularly with Yang Gang's group, and then kind of next steps. So what are CECs? The Environmental Protection Agency defines them as pollutants for which there are limited data on occurrence, environmental fate, and toxicity. Generally, they're thought to include things like PFAS. And I won't pronounce those names, but I think you've all probably heard about them. Pharmaceuticals, phthalates, which are plasticizers, which are ubiquitous in the marine environment, personal care products, UV filters like sunscreen, whatnot, flame retardants, and a whole variety of other things. And these are in addition to what we would call contaminants of known concern, which include, in this case, a lot of banned pesticides like DDT, PCBs, radionuclides, PAHs, which are derivative products from oil, toxic metals, et cetera. So there's a whole cocktail there of different pollutants that are in the environment. We're going to focus primarily on the CECs, but along with all the chemistry we're doing, we're actually getting some really interesting data on these contaminants of known concern, and particularly the ones that have been banned for decades, right? They're still there. So we have 3 funded projects, and they focus on various aspects of CECs and the legacy pollutants that we're calling. And it's based on an initial pilot project that we did between 2020 and 2022. And Chris was talking about, going out on the boats or not going on the boats when, you had a problem with COVID. So a lot of that started on my personal boat catching fish by myself, right? So anyway, we published a paper based on that, and it really set the stage, I think, for justifying a much more expansive project. And then the project is 5 years in duration. It started in 2023. And so we've got these different components, which I'll explain. The project itself, the Barnacle Baseline Project, which is funded by South Carolina Sea Grant, and then what we're calling the Tampa Bay Reach Project. And I can't remember what that acronym actually means. There you go. Okay. Perfect. You can see it's fishing based. So our objectives are, number 1, to sample the sediments, the oysters, the barnacles, the fishes, and the fishers, right, of Tampa Bay, to characterize the concentrations of these different pollutants, to ascertain the relationships between contaminant body burdens and what is the, for example, the health of the fish. And we can do that by looking for symptomology in terms of liver tumors and cancers and those kinds of things, to characterize the associations between contaminants. And interesting thing is, with this long list of contaminants, we know that the mix of those contaminants is different around the bay. And so what we want to do, through Josh Kilborn and others, is to look at the pollutants in a multivariate space to try to see if there are associations in different parts of the bay that are characteristic. So for example, the heavily urbanized part of the northern bay is probably going to look a lot different than the agricultural parts of the lower eastern side of the bay. And so trying to understand from the fish's point of view, for example, or the people's point of view, not only one contaminant at a time, but also the totality of the mix of chemicals that are in the animals. To survey the human consumption patterns, focusing particularly on subsistence level fishing, and I'll explain that in a little bit. But this is important because there are many people who rely on the protein that they derive, and their consumption frequency is much higher than the average population in the United States. And this goes into risk assessment models about how frequently you eat things and what the contaminant levels there in there. And so it's very likely, based on a lot of historical evidence, that subsistence fishers in particular are at much higher risk for contaminant level effects. To develop quantitative risk assessment models for seafood consumption, which are based on some of the surveys that we want to do. To test prototype remediation methodologies. People talk about, what the problems are with PFAS and other things, but what could we actually do about it? And so Maya and her student AJ have been working on that. And lastly, to try to keep in contact with the counties, the state, the federal regulators in terms of what's going on, because there's very little regulatory oversight of some of these chemicals. And in many cases, they're not even established in terms of the action levels, for these various things. So the more we can actually shed light on the impacts of these chemicals, then these regulators can actually use the information. So the previous study we did on PFAS, 2020 to 2022, we did both sediment grabs, and you can see here. Oops. Yeah, okay. So these are sediment grabs in various areas of the bay. This is Old Tampa Bay, McKay Bay, which is that little appendage to Hillsborough Bay, the mid Tampa Bay areas here, lower Tampa Bay, and then Terracea Bay, which is this small bay down here. And you can see in the sediments, very high levels in Hillsborough Bay and Old Tampa Bay, weird that McKay Bay wasn't, because there's a Superfund site right there, you know, and so if you look at the fish, they actually quite contaminated. So you see this decline of, in this case, PFAS, you know, very high levels in the northern part of the bay, and they go down. So this was revealing to us. One of the interesting things is if you look at a chemical called PFOS or PFOS, that's one of the major constituents of PFAS, right? Which is a number of chemicals. The drinking water standard by EPA is 4 parts per trillion. And in the case of some of the fishes that we got, it was up to 31,000 parts per trillion. So it's very clear that, you know, there's a concentration factor going on as you like move up the food chain. Okay, so in terms of CKCs, so these are the contaminants of historic interest. And so NOAA has a mussel watch program, which they basically looking at oysters throughout, you know, the country, and in particular the Southeast United States. So they have 6 sites in Tampa Bay, and so they've been doing their chemical analysis since the 80s. Since the 80s, right? And so it's really interesting, because if you look at some of these chemical mixes, these are insecticides here. This is DDT, and it's metabolites here. These are some of PCBs, and you can see that DDT was outlawed in 1972, right? So this data set starts in 86, and the total concentrations in oysters has gone down significantly. Same thing for these insecticides. Mirex, for example, was used to control fire ants. You know, so there's a lot of spread of that, and you can see those chemicals are going down. These are the years when they were banned, and this is PCBs, and you can see the totality of PCBs has also gone down. So that's really good. You know, I mean, they're not gone, but you know, they've gone down. So the issue is whether we've replaced one set of chemicals with another, right? And so that's part of the research that we're trying to do. So the project, in terms of project components, the first one is sediments. So this is basically a collaboration between, you know, very familiar names at Eckerd College, their students and interns, and then Isabel Romero, who many of you know, used to be here, now is a prof at the University of South Carolina, and her student. So what we're basically looking at is a combination of sediment cores and also grab samples. And the sediment cores, obviously, if you've got an undisturbed core, it becomes a chronology, right? So you know what was there 20, 30, 40, 50 years ago, and what's there in contemporaneous sediments. And that's the ideal, to actually understand the dynamics of these various pollutants, if in fact they're conserved in the sediments, much of which is anoxic, right? And so we've done a variety of sampling on all sorts of different vessels and using multiple techniques. So here's where we've collected sediment samples. Our goal is at least one chronology from each of the 6 sub areas of the bay. And we want to pair those chronologies with measurements of the contaminants at various levels. And of course, we can derive ages where those samples come from the radionuclide degradation over time. So these are really important. The trick is to try to find an undisturbed area, like it hasn't been dredged, or it, you know, the heavy weather that we get, you know, hasn't resuspended the sediments, etc. There's some really good cores, you know, particularly up in old Tampa Bay, you know, up here and over here, and then surprisingly over here near the channel, you know, for the shipping channel. So those are in process now. So the second component of our project is the benthic invertebrate component, and we're focusing on 2 sets of species. American oyster. This is an oyster reef in McKay Bay that we were sampling a couple of weeks ago. Obviously, for oysters, there's a lot of consumption of oysters, even from Tampa Bay. There are oyster leases in the lower part of the bay, but also we know that from subsistence harvesting that people eat these as well. And then also we're focusing on barnacles. And the reason we're using 2 different species here is barnacles, obviously they're not consumed, but one thing you can do is that they can recruit to, different substrates that we put out there. So we know the entire life history of them as benthic adults. And so if you look at the contaminant levels over a period of time, you know exactly how long they've been there. And so as opposed to sampling the oysters from the natural reefs. And two students here, AJ Gross is doing the oysters and Lane is doing the barnacle project. Okay, so oysters in Tampa Bay. So if you look at the blue dots on this graphic, you can see that this is a survey that Florida Fish and Wildlife did in 2020. So they're all over the bay, right? So they're an easy target. The stars represent the places where NOAA has been doing their mussel watch program, right? So we tried to actually both do the oyster project and the barnacles in association with that. So we'd have some historic context for the various contaminants of known concern. And so for the oysters, the design here is there are 18 oyster sites outlined in the red, over 4 quarters, 2 subsites per sample, a total of about 1,200 oysters or so to be sampled. And so we've done the first quarter and on to the second quarter. And so it'll be interesting to see, number one, if we've got regional differences in the pollutants and what they are, but also if we have a temporal difference in pollutants based on the flux of wet season versus dry season, for example. So on the barnacles, again, this is a project that's funded by South Carolina. They have sedentary adult stages, but they have pelagic larvae. They recruit and grow very fast. And again, we're focusing on regional and seasonal differences in both the classes of pollutants. So the deployment of the barnacle collectors is very similar around the bay. Several of them are deployed near wastewater treatment outfalls, particularly that one, which is the big one in Tampa Bay. It's about 50 million gallons a day that come out of that wastewater outfall, and we have a barnacle collector right next to it. So it's an interesting story. So this is Lane pounding one of our collector arrays in. So the array is a 3x3 set of tiles. And the reason why we're using glass tiles, is that they're inert to the chemicals, right? And so a lot of times people do recruitment studies for benthic invertebrates, but they use a substrate that itself has pollutants in it, like PFAS, right? So we're trying to basically keep those clean. So the collector plates have a temperature gauge on it. So we're basically collecting temperature every 5 minutes, right? And then also it has an air tag on it so that we can keep track of them. That's an interesting story. So we have didn't know we were still hammering. We put a test array out in the backyard, off the finger pier where the tokobog is. And so this is week 2, and you can actually, if you squint, you can sort of see barnacles. Week 4, week 7, that's week 11. So barnacles really do recruit. I mean, and these are massive barnacles. I mean, if you look at that little red circle, that's them. And so, yeah, the whole thing works, in terms of collecting things. And so this is a screenshot of the air tags that we've got on the thing. So the air tags are located along the top bar here. So at low tide, the top bar comes out of the water. And so if anybody's driving by with an iPhone, you'll get a hit, right? So some of them hit an awful lot. That one down there there's lots of fishermen down there, so we see it all the time. Right? Some of them, we know they're there, but what's amazing to me is how little vandalism has happened there, because we've been out to these sites. Go figure, right? So on the fish side, we have 4 target species: snook, red drum, spotted sea trout, and sheep's head. So the sampling design was sampling quarterly. We wanted to sample the 6 areas, and the goal was to sample 10 of each species in each region for an entire year. We started that in April last year, and we completed this fish work in April of this year. You can see where all the fish are coming from. One of the things is I now have all these numbers, that our captains, took us to. So I know where all the fish are. If you could come by for, some drinks, and maybe we'll talk about that. You know, so we're using basically recreational charter boats to do this, and much more efficient than us trying to figure it out. So this is some of your colleagues catching various fish around the bay. The biggest, longest fish, the biggest fish. There's AJ, big red fish. I can't say it was a lot of fun. It was so these are the size composition data for all the species. And the species are regulated by a minimum and sometimes maximum size. And so these are the so-called slot limits, where these are all undersized fish. We catch a lot of undersized fish, but that's okay, because what we're trying to do is to see if the little ones have lower levels of contaminants than the big ones, and that they're accumulating them over as they grow to larger animals. And also we know that when people catch these fish, they don't necessarily obey all the rules. And so if people are basically catching and eating a lot of snook that are, say, 50 centimeters, we ought to know that and factor that into what we're doing. So quite a few fish. Okay, so in terms of what we're actually doing with the fish, after we catch the fish and have them on ice overnight, we basically have just dissected them all. And so all of these tissues are basically sampled as part of the protocol. The otoliths are used to age, and all of those fish that we caught have been aged. Good job. And then we're also focusing right now on the muscle because of the consumption issue, the livers, because that's the way that animals detoxify, and also the gonads because of a study looking at potential for parental offloading of contaminants, right? But we also have another study looking at vertebrae to look at radionuclides, and then we've archived the stomach samples, which I think are going to be really important in our long-term studies. The questions we have are, a lot. Are the relationships between contaminant concentrations and fish biometrics, age, size, sex, etc.? Those are important. Are there spatial and temporal variations in contaminants? Now, fish move around a little bit, right? They're not like the benthic invertebrates, but nevertheless, our pilot study showed that, we had higher concentrations of the fish in the northern part of the bay. So is that a function of we sampled different species or what? So concentrating on the same species, but understanding, it from, the spatial and the size variations will kind of sort that out. Do the contaminants accumulate and or do they biomagnify? That is, if one species eats a lower trophic level, do you take on all the contaminants and incorporate them in your body? And then lastly, is there this issue of parental offloading? Parental offloading is seen in a number of species, in particular marine mammals, where the mothers, when they give birth to the calves, they basically concentrate all this lipid in their milk, and then they basically lactate the calves, giving them all the pollutants, because the pollutants are lipophilic, right? And so we're trying to look at that issue. So Alexandra had a poster at the American Fisheries Society meeting a couple of weeks ago. She won Best Poster Award for that, looking at the lipid budget in these guys between the liver, which is like a battery, and the gonads, which are like consuming the lipids as they mature. Really interesting study. Right. So one of the last big components we have are the Fisher surveys. We talked a lot about animals and what was on there, but in order to get a handle on this issue of subsistence, we've got a significant amount of effort going into looking at the social side of this. And so Heather O'Leary, who's an anthropologist, who many of you know at USF St. Pete, as well as Noel. Okay, yeah, right. And then Jesse Fly, are leading this, and we're using a lot of the Eckert students for that. So they're asking each of the fishers, and we've been sampling, these are the sample sites that we're trying to look at. There's these 40 questions. Well, there's really kind of 3 questions I wanted to focus on. One of them is, are you eating the fish that you caught? And then how many times a week are you likely to eat these, or how many times a month? and then what species do you actually catch? And the data are really fascinating. So for example, how many times fishing have you spent in the last month fishing, right? They go from basically 0 all the way up to 20. So some people are out there 20 times a month, doing subsistence fishing. And these are the number of times that you're eating the fish you caught. And it goes, the average is 5 times a month, but it goes up to 30 times a month, right? So basically some people are eating the fish they catch out of the bay every day, right? So those are an at-risk population, right? Really fascinating data. And then in terms of what are the top 5 species that you caught, not necessarily in the bay. These are really interesting because all of the species that we talked about in terms of being our, target species are there. Redfish, and there's some different names for redfish here, snook, trout, and then sheepshead. And then also some other things like, I went fishing for tarpon. Well, nobody eats tarpon because it's a prohibited species, but it's really interesting to look at this because there are some species on here that don't occur in Tampa Bay, right? So clearly what's happening is they're integrating all of the fishing they do like in freshwater, et cetera. So we might want to refine the question a little bit. Interesting stuff. Okay, so the other part of the social science interview process is looking at different groups in terms of their perceptions of what they won't eat, as opposed to the ones that are catching. And so this was a Wordle based on interviews of nearly 450 people at the GomCon conference last February, right? February 24th. And so clearly people think that oysters are polluted, right? And they don't want to eat them. But there's some other stuff, none, et cetera, et cetera. And then this was some interviews of the captains from the King of the Beach fishing tournament. These are people that fish outside the bay, right? So they're fishing offshore. Catfish is the big one, right? I presume they mean gaff topsail catfish, which is a catfish. It's a sea catfish that's there and it roots around in the mud, right? So I wouldn't eat them either, right? So, but anyway, so it's really interesting because, some of these things are like none. I wouldn't eat any fish out of the bay, et cetera. Lots of different things going on. Really fascinating. And I do think different perceptions based on who you are and what you do. One of the last things I want to talk about was remediation. And so this is the outfall from the Curran plant. This is actually, you think about a wastewater treatment outfall pipe. There is no pipe, right? It's an underwater pipe, right? And so all you see is this huge whirlpool, that's around there. And there's 2 danger signs, don't go in it. When it's flowing, it's a dangerous place to be. So we actually put our collector plates right next to it. Right? Comment on that. And so this is work that Maya and Isabel and AJ are working on. And so what they wanted to do is look at a process called electrocoagulation, which is, could you take the wastewater, from the treatment plant and run it through basically an electrolysis kind of process where you've got, basically the anode and cathode, and will it actually remove in the flock the pollutants, right? So they did a number of samples based on wastewater from the South Cross Bay wastewater treatment plant. And it's really interesting. And so here's a bunch of, they're primarily pesticides. And this was the raw effluent coming out of the plant. And this is the treated effluent going through the electrocoagulation. And the interesting thing is I'm highlighting DDT here. You can see that DDT concentrations in the wastewater effluent are about 1 part per billion, which, that was banned in 1972, but it actually took it down significantly. And this basically shows that this process actually works. Whether or not it's economical to actually process 50 million gallons a day, I doubt it, but clearly there are processes that one could potentially use. Yeah. So some thoughts on that. Electrocoagulation actually works. Obviously they tested iron and aluminum electrodes. The iron seemed to be more efficient, and the interesting thing is these CECs and CKs are numerous, and they're concerning because banned pollutants from years ago are still showing up in basically the water you drink. So last a little bit. That's okay. Okay. Last little bit I want to talk about is systems analysis and modeling. One of the things that we're very interested in is how do pollutants get into Bay, right? And where do they go? And so if you look at, where pollutants come in from, they come in from land-based sources, that is, non-point source runoff. They come from atmospheric deposition. And then, obviously wastewater outfalls, and we talked about that today, and then probably groundwater intrusion, right? So understanding that, and also, they exit the Bay too through the mouth. And so understanding the mass balance of pollutants, I think, is going to be really important, particularly as it relates to this sort of sediment well, and so the other aspect of this, of course, is the biological system, and, focusing on oysters and then their consumers, barnacles and their consumers up to humans. This represents a really interesting system to try to model. And so number one, understanding mass balance of pollutants that enter and exit the Bay is going to be a priority. Secondly, we want to do some particle tracking of the fate and simulated inputs from the wastewater treatment outfalls, and using Bob and Yonggang's model, and it would be an ideal tool. And lastly, we want to do some food web and contaminant flows. For example, if we know the pollutant level in the oysters, we can figure out how many oysters, say, a redfish eats. Same thing with barnacles and sheepshead, and then look at the biomagnification and pollutant tracking through the ecological system. This is work to be done. I just wanted to emphasize the use of the hydrodynamic model. So this was a study that Yonggang and Bob and others published from the Piney Point thing. And so the Piney Point the discharge was here. And so this was, sort of late in the Piney Point thing. You can see the pollutants traveled primarily in the mid bay, but even got up into the lower bays, but a lot of it exited as well. So our idea is to basically take these sources, which are the wastewater treatment outflows in Tampa Bay, and simulate them as well. And probably in proportion to the number of gallons that each one of them is contributing to it. So if, say, for example, we're trying to track PFAS from wastewater, we ought to be able to understand how they accumulate in the different parts of the Bay. And that may relate to the concentrations that we're seeing in the biological system, the benthic invertebrates and the fish. So that would actually give us some way to track that. Okay, so our next steps, obviously we want to complete the field sampling for the benthic invertebrates and sediments, complete the GCMS analyses for the pollutants and all the biological and sediment samples. We are challenged about the interviews. We had a NSF grant that was supporting that, but that got terminated. But Eckerd College came through big time. And so we're going to keep the samplers out there for a full year, which I think would be really valuable data in terms of understanding that. And then do the seafood risk assessments. Want to start to look at how we would do the systems analysis of this and the modeling studies. And then we're, like I say, we're good to 2028. So hopefully we can make that all work. And then I really want to thank our field samplers. Aaliyah is here. There she is. Our charter captains and the technicians who kind of making this whole thing work. So thanks.
As I was saying, before I was at GNS, I did my PhD at Victoria University of Wellington, also in New Zealand in the Antarctic Research Center, working with Nick Gullidge and Nancy Bertler. Before that, I was a professional distance runner for the Boston Athletic Association. So
No, it's no, it's then you can
okay. Yeah, okay. But I am happily retired from that. So you can invite me on a short jog, but that's about the extent of it. I used to work in turtle conservation. And yes, that is a photo of me putting on a turtle costume to entertain some kindergartners. And I do have a labradoodle that is making the long journey from Auckland to St. Pete in the not too distant future. So, oh, wrong way. So today's talk, onto the science. I'm going to briefly go over sort of some past work that I've done in the Ross Ice Shelf, talk a little bit about New Zealand's Antarctic Science Platform, which is the main funding mechanism for Antarctic research in New Zealand. I'll talk about the proposal that we have under review. And then lastly, just a slide or two on what I'm thinking for a new research group at the College of Marine Science. So, you know, why the work I do matters. Ice sheets, very important for sea level. Antarctica has this really important connection to the ocean, and that's because of the ice shelves that fringe the continent. You can see here in this plot the ocean thermal forcing ranging from low in a yellow color to high in a green color. And you can see next to where the ocean thermal forcing is high, you have these really high basal melt rates of the ice shelves. And why this matters, ice shelves buttress all of the land-based ice upstream. So if they start to thin or become unpinned, you increase that flow from ice to ocean, that raises sea level. The Ross Ice Shelf, where I'm going to be talking about today, is quite interesting in the sense that it's not really melting all that much at the moment. It's what's called a cold ocean cavity. You can see there are kind of three different modes that we think about ice shelf melting. If you ask a physical oceanographer, it's much more complex than this, but for the purposes of this talk, I'm going to go through these modes. Mode 1, which is where, similar to the Ross Ice Shelf, you have this strong production of dense shelf water. It's very cold. It's associated with relatively low melt rates. You have mode 2, where you get this intrusion of warm circumpolar deep water into the cavity, and that causes very high melt rates. And that's what you see in the other parts of West Antarctica, where you see all of that red. And lastly, mode 3, also important for the Ross Ice Shelf, you get subduction of warm Antarctic surface water that melts the ice shelf front. And so while the Ross Ice Shelf has very low melt rates today, we know that in the recent Holocene, that might not have been the case because there's radiocarbon present in subglacial till. This study in Nature found that it was post-LGM in age, but they weren't able to precisely date it. Using an ice sheet model with a relatively simple glacial isostatic adjustment scheme, they determined a plausible mechanism is that as the ice sheet retreated out of the last deglaciation when the bed was much lower, crustal uplift regrounded the ice sheet back to its present day position. But we know from work from Ryan Venturelli and Brad Rosenheim using ramped pyrox, which is a more accurate method of dating particularly low carbon sediments. They were able to date the timing of retreat beyond the present extent at the Whillans grounding zone to be between 7.5 to 5.3 kiloyears before present. This is much later than that was indicated by that original study, and it's also more consistent with ice thinning records from the Transantarctic Mountains. So my recent work published in Nature Communications that I talked about when I visited a couple of months ago, we used a coupled ice sheet global glacial isostatic adjustment model that had various Earth structures. What we found is that the best explanation to retreat the ice sheet beyond the present day position within this time window suggested by the proxy records is a transition from that mode 2 melting where you have warm circumpolar deep water getting into the cavity to the present day mode 1 melting of dense shelf water production. But what we also found is that the solid earth is still really important. And in particular, the mantle viscosity controls that rate of ice sheet response from that change in ocean forcing. This work that I did was part of New Zealand's Antarctic Science Platform. The New Zealand government understood that for Antarctic research, particularly with field components, it's really helpful to have that long-term security of funding that allows our logistics partner, Antarctica New Zealand, to be able to plan events, but it also really helps for seeding those international collaborations. And so what they did is, we had still go through a peer review process but we had these 4 different projects, but the funding itself was guaranteed. And the overall goal of the program was to understand Antarctica's global impact on the Earth system and how this may change in a warming world. And so my work was part of Project 1, which was focused on ice sheet dynamics. The aim was to investigate the past and current drivers, mechanisms, and feedbacks of ice sheet retreat to inform our future projections. And then I took over a leadership role in this program from 2023. So we did a lot of different things as part of this program. In the talk that I mentioned a couple of months ago, I talked about our SWACE 2C program, where we're actually drilling for sediment at the CAM grounding zone. We also did a whole heap of modern observations of ice-ocean interactions. We had an ROV that was exploring basal crevasses in the grounding zone. We've developed new regional climate models of atmosphere-ocean coupling, ice sheet-ocean coupling, and ice sheet solid Earth coupling. We contributed to the International Ice Sheet Model Intercomparison Project for CMIP6. We reconstructed the deglacial history of the Bird Glacier, and then we had a whole heap of observations, of geophysical observations across the Ross Ice Shelf. Thankfully, the New Zealand government has renewed the program for the next 7 years. So this past year has been a lot of development of what the next phase looks like. The new director, Natalie Robinson, has developed a new framework for it that's kind of inspired by a traditional Maori waka, which is a Maori canoe. We have 2 main projects that are our marine navigational guide, Tiaki Moana, and our ice sheet-based project, which is Tiaki Whenua. And then we have this central question that's motivating both projects, which is what are the critical signposts of catastrophic climate change and how can we effectively observe them to support timely mitigation? With that concept of signposts of change in mind, our team quickly settled on a key location for the Ross Ice Shelf to study for long-term monitoring as the Cape Crozier region, which is adjacent to Ross Island. It's where we have the highest heat flux into the ice shelf cavity. The mechanism is that mode 3 melting, where you get southward Ekman transport of warm Antarctic surface waters, and that melts the front of the ice shelf, and you have much higher melting relative to the rest of the ice shelf. It's also an important area glaciologically because it's a key shear zone at an ice shelf pinning point. And then adjacently on Ross Island, it supports a large seal and penguin population. So it is an opportunity to study the ice, study the ocean, and study some of the ecosystems as well. And so our main targets were long-term monitoring of this southward Ekman transport, understanding the seasonality, which I'll get into a little bit, understanding how rifts form in the shear zone, and then understanding the population dynamics of mesopredators. From a glaciological perspective, this is a study that was done as part of our first phase of the program, which highlights the importance of this region. These are model simulations that show that effect of changes in basal melting to ice above flotation. That is what contributes to sea level. And what we see is anywhere with dark purple is a sensitive area to a change in basal melting. So this Cape Crozier corridor that we're targeting is important for Antarctica's future sea level contribution. There are really strong seasonal changes that make it an important region to study to understand changes in the melt regime. This is a study from Craig Stewart, now at Earth Sciences New Zealand, showing in the top panel a change from a southward directed flow in the summer, which shifts in the wintertime. And in the summer, you have the breakout of the sea ice, which causes this big spike in sea surface temperatures. related to that solar absorption when you no longer have the sea ice. And what this results in is every summer you see this big spike in melt rates that more than triples. And so getting that seasonality, this is a really good place for us to understand those mechanisms of basal melting, because you go from, essentially very low melt rates to very high melt rates. Why this is particularly important is basal melting is a key source of uncertainty in our ice sheet models. This is an example that a lot of the state-of-the-art ice sheet models use as what's called a parameterization. It essentially takes your conditions in your climate model out on the continental shelf. It simulates your melt at the grounding line, and then through a box model system, you might have some refreezing at the ice shelf front. And as you can imagine, in the Ross ice shelf, that's probably not a good parameterization to use when you are getting a lot of your melt in this area. The bottom panel shows results from the ISMEP6 extended runs to the year 2300. What you can see is ice sheet model simulations and their simulated ice shelf melt for different climate models running out under a high emission scenario. And what you can see is there's tons of uncertainty, and this is more related to the ice sheet models than what's feeding into them. And so this is a really important area for us to target through an observational perspective. And so in April, New Zealand announced a strategic fund of up to $3 million to support collaboration between New Zealand and the US for Antarctic research. Expressions of interest were due in June, and from there, a smaller pool of projects were selected, given a little bit of funding to put in a full proposal that was due on August 18th. I do have an asterisk because the NSF has not fully confirmed that they are going to participate in this program, so we don't know what the US side of that funding looks like yet. But there is an expected announcement in September, one way or the other. Regardless, the successful proposals will be notified in September and commence on October 1st. So what we have in mind is the Cape Crozier Heat River Expedition, or CORTEX. We have 4 aims for the project. The first is to identify and quantify that warm inflow, both its rotational effects, its seasonal structure, what that means for basal melting, both through ship-based hydrography and also borehole deployed ROVs. We've worked in the past for a long time with Brittany Schmidt's group at Cornell, but now we've also connected with the JPL Ice Node team. You can see the figure in the bottom is the ice fin in the McMurdo Sound. The second aim is to determine the influence of ice shelf basal roughness on the turbulent transfer of heat from ocean to ice using autonomous phase sensitive radio echo sounders, or AP-RES. The third aim is to understand rifts, observe conditions that drive ice shelf rifts to form, and determine their oceanographic influence using a new autonomous profiling system from the Scripps Institute of Oceanography, and then also those same ROVs that can explore basal crevasses. And then lastly, I think the hardest job of all, and the one that I would be leading, is to integrate all of these novel observations into large-scale ice flow models. And so this would be a collaboration with some of my colleagues at Victoria University of Wellington and the University of Otago. This is a look from Craig Stevens, who is leading this proposal of the field site. What you see is the planned hydrography that would be part of the next phase of the Antarctic Science Platform through the course of 3 voyages. And then in orange, this is where we plan to install a few new boreholes to study those ice-ocean interactions. A cross-section view is shown in this panel. You can see we aim for a long-term mooring can withstand multiple years, so we can get multiple seasonal cycles, and then also using the ROVs to explore the basal crevasses, the rift sampler from the rift docs team to understand it from above and understand the role of melange in rifts. And then lastly, using acoustic source to better understand that turbulent transfer of heat from ocean to ice. The model integration part of the proposal, we've identified two very different ice sheet models. The first is the model I work with, the parallel ice sheet model, and then the second is the bicycles ice sheet model. They're very different. PISM has a uniform grid. There's a sub-grid parameterization for the ice sheet grounding zone, whereas bicycles has an adaptive mesh, which means as the grounding zone changes, the resolution changes with that to focus that high resolution to better resolve those features. PISM is initialized by a spin-up procedure. This allows you to do paleo simulations. In contrast, bicycles is initialized through data assimilation. That gives you really accurate ice sheet geometry at the start of your simulation, but you do have to make a number of assumptions about the internal ice like thermal profile, and then that can sometimes lead to quite significant model drift. PISM has a calving law that's applied, whereas bicycles has a fixed calving front. You can only lose the front of the ice shelf through basal melting. PISM's relatively more efficient to run, less so for bicycles. And so the idea is that we can improve basal melt parameterization in both types of ice sheet models. We plan to do high resolution, which is 2 kilometers or finer. In the case of bicycles, even up to 100 meters in that Cape Crozier corridor for the Ross Ice Shelf catchment. We can compare our simulations using a few different basal melt parameterizations to our multi-year observations, and then do some model development to improve that. And then lastly, we can do some scenario-based experiments. What happens if that seasonal signal, that high melt event occurs for a longer duration during the summer? What if you get Antarctic surface water intruding further into the cavity, or even circumpolar deep water? This is a look at timelines. So the top is what is currently in the Antarctic Science Platform. That's already been through review and is being contracted as we speak. We have our extension of our AP-RES network across the Ross Ice Shelf, with the aim for our Crozier camp at the end of the program. And we also have 3 voyages scheduled during the course of the program. Everything in the bottom is through this new Cortex US-New Zealand collaboration, including our 4 different research aims. We would aim to test some of the ROVs in McMurdo Sound sea ice before moving on to sort of the rift and borehole works. But this additional funding essentially allows us more years of doing those borehole experiments, and also would support a lot more modeling as well. With this in mind, the type of group that I am wanting to start here at the College of Marine Science is focused on ice sheet interactions with the soft and solid earth and the ocean. And I'm tentatively calling it Ice 2.0, as in ice sheet flow to ocean. So we're really interested in what are the global consequences of ice sheet dynamics and melt. And so we'll be applying numerical modeling and machine learning techniques informed by geophysical observations and paleoclimate records. I have a website under development. It's, I think, nearly ready, but you know, it essentially has projects that someone can click on, a prospective student, find out a bit more, and contact me. And that's it.